ಇದನ್ನ ಅಲ್ಲಿ ಅವರು ಕಂಟ್ರೋಲ್ ಮಾಡ್ತಾ ಇದ್ದಾರೆ.
Yes, sir. Now we are online, ma'am.
Yes.
Good morning, everyone, and welcome to this webinar on mathematical models on epidemiology in connection with COVID-19. First of all, I'll let you invite our Dean, Professor Mary Saral, to say a few words on this event. Ma'am, I'm sending you live, ma'am. Yes, ma'am.
Yes. So very good morning to one and all present here. It's my pleasure to welcome you all for this webinar on mathematical models on epidemiology in connection with COVID-19. Before I could give my inaugural address, it is my duty and also pleasure to welcome the participants, Professor B.K. Mishra, Principal, Markham College from Hazaribag, and Professor P.S. Datti from Bangalore, Professor Anuj Mubayi, Associate Director from Arizona State University. So I welcome you all for this inaugural schedule as well as the webinar. And indeed, it is my pleasure to have you all here on this online platform where everybody is afraid and frightened of COVID-19, and we are there to discuss about this COVID-19 here. And even I was surprised when my colleagues from Department of Mathematics came forward in saying that they would like to organize this webinar, which is related to COVID-19. And there isn't response from physics and chemistry, but mathematics faculty members have came forward to think about this and to create an awareness about this deadly disease, the pandemic situations, as well as I'm happy that all of you are very much interested to give your inputs how to manage these pandemic diseases. And this is truly outcome of interdisciplinary research which is happening in School of Advanced Sciences. So I would like to brief you about the Department of School of Advanced Sciences. So we have Department of Mathematics, Department of Chemistry, and Department of Physics with us. And there are about 230 faculty members in School of Advanced Sciences. So collectively we call it as a School of Advanced Sciences, which is one of the biggest school in VIT University, rather VIT Institute, I could say, and we are leading in our research. So our faculty members are very strong in research, and half of the strength of the school is shared by Department of Mathematics. So more than 115 members are now with the Department of Mathematics, and they are doing excellent job in organizing various events as well as encouraging the students to participate the activities even during this outbreak time. So we have conducted various programs like additional learning on mathematics and learning mathematics while at home. So these are the various activities being organized by Department of Mathematics. So at this juncture, I would like to congratulate the head of the Department of Mathematics, Dr. Kumar, and the coordinator of this event, Dr. Is that Ashish Kumar, right?
Prasad.
Ashish Kumar and Prasad for having coming forward to organize this event. So hats off to you all, and also the other department members from Department of Mathematics. I think they will be very happy to coordinate, and they should be on this platform to listen to all. And I'm happy that the speakers came forward to participate in this webinar. So wish you all happy learning through this webinar, and also I congratulate the participants who have registered for this webinar, and I wish this webinar to be a successful webinar. And thank you so much for giving this platform to meet you all through online, and I wish you all the best. And once again, on behalf of management of VIT, I would like to express my sincere thanks for accepting our invitation and to share your knowledge with us. Thank you all. Thank you, sir.
Thank you. So, Prasad sir, voice is not audible.
Yes, sir. Now I send you online, sir. Now you can speak, sir. Then.
Yes, sir. Good morning, everyone, and respected Dean, Madam, and learned resource persons, Professor P.S. Datti from TIFR, Center for Applicable Mathematics, Bangalore, and Dr. Anuj Mubayi from Center for Collaborative Studies in Mathematical Biology from Arizona State University, and Professor B.K. Mishra from BITS Mishra. And I thank on behalf of the Department and School of Advanced Sciences I thank the resource persons for accepting our invitation to be part of this webinar and sharing your expertise. So as our Dean Madam rightly mentioned, so I'd like to congratulate our organizers of this webinar, Professor Ashish Kumar Prasad and Professor B.S.R.V. Prasad, and for taking the initiative to host this relevant topic of the present day need, mathematical models on epidemiology in connection with COVID-19. Everyone is caring about the COVID-19 and the consequences, but as a mathematician and with the mathematical biology, connected with the mathematics and biology, we have to bring it a model.
Sir,
So, sir?
Sir, I think 1 minute, sir. There is some problem. Technical error.
Yes, sir. I think it
Yes,
is okay, sir.
now it's okay, sir. Yes. Yeah.
Okay. Sir, at the outset, I'd like to thank our Dean Madam for encouraging us to do this type of activities in the School of Advanced Sciences and also in VIT. So I thank the resource persons, Professor P.S. Datti and Professor Anuj Mubayi and Professor B.K. Mishra for accepting our invitation and sharing their expertise related to mathematical models and epidemiology in connection with COVID-19. I hope all the participants, we have around 440 participants from different parts of India and also from abroad attending this webinar. I'm sure you will be benefited from this webinar from 10 to 1. So I thank this opportunity and I thank our VIT management for providing us excellent facilities. Our Department of Mathematics consisting of 115 faculty and our School of Advanced Sciences is one of the leading schools in VIT in terms of research and also in teaching. So with these few words, I thank the VIT management and the resource persons and our dean, and I congratulate the organizing coordinators of this event. And I also appreciate all the participants for your enthusiasm to learn the new dimensions in mathematical modeling. So thank you, sir. Please go ahead with the schedule, sir.
Thank you, sir. Sir, Professor, I'm Anuj, Professor Anuj. I'm sending you live. The one thing, just your introduction is going to be given Ashish. Then you can start your presentation.
Sure.
Okay. So I'll request Professor Ashish to give his introduction of Professor Anuj.
Welcome, learned professors, researchers, and dear students. COVID-19 is knocking our doors. More than 15,000 persons get infected in last 24 hours in India, which is third highest after Brazil and America. So it is necessary to help our nation to fight against corona. So we have here, first of all, Dr. Anuj Mubayi. Anuj Mubayi is an applied and computational mathematical scientist whose research is driven by the mathematical modeling of problems of interest to the public health or social science community. He has extensive experience in the health decision analysis and in successful development of a strong mathematical sciences training program. He is an associate director at Precisionheor and held faculty position at Simon A. Levin Mathematical Computational Modeling Science Center at the Arizona State University, Temple. He also co-directed Mathematical Theoretical Biology Institute and was earlier Associate Research Scientist at the Prevention Research Center, Berkeley. He is a research member of the National Alliance for Doctoral Studies in the Mathematical Sciences and the Intercollegiate Biomathematics Alliance. So I request Dr. Anuj Mubayi to start his talk, and I request all participants to give their questions on questions and answer blog. So they can post their questions and Professor Prasad will inform Dr. Anuj Mubayi about their questions and he will answer your questions. So thank you. So Dr. Anuj Mubayi.
Anuj, can you share your presentation, Anuj?
Yeah. So I will switch off my video because just to have the better quality of the presentation. So can you see my this thing?
Yes. Yes. Okay. I'm sending live.
Okay.
Yeah, it's online now.
Okay. Thank
You
you.
can go full screen. Yes. Yeah, we are able to see.
Okay. So greetings, everyone. Good morning, Tuesday. Good morning in India, and it is evening here, late evening here in US. It's my pleasure and honor to speak. I was invited by Dr. Prasad, and it is always, I know him for a long time, and this was, I mean, we chatted on various things in the past, and it's really pleasure and as well as honor to be part of this COVID-19 modeling seminar that he and his collaborators have organized. With that, I would like to first say, give my gratitude to Dean Mary Saril for telling me so many things about VIT I was not really aware of. It seems like an amazing institute. I may have to apply for a position there at some point of time. And also gratitude to Professor Rishi Kumar as well as Ashish Kumar. They are all organizing is not an easy job for especially an event like this one where so many people remotely are being joining to this this webinar. So with that, let me give what I would be talking about today. Before I go into the details of it, I want to be very clear that I'm not an epidemiologist. I'm not a clinical expert. I'm only a mathematical modeler and help understand disease better and study control and evaluations of the control program. So such as social distancing, basically. So my training has been, my PhD is in applied mathematics, almost more than 10 years ago. I had an experience working in Department of Infectious Disease in a hospital. I also was part of a clinical trials before. And currently I'm part of organization called as Precisionheor, where I'm associate director. And we work on health economics problems and evaluating control programs and cost-effective ways, suggesting some of those policies, basically. So with that, I will go further. You know, I was thinking what would be the best strategy to talk today. I just 2 days ago gave another webinar, and so kind of out of ideas, but eventually pulled out together some of the things that is very relevant to COVID-19, but also give you understanding about how, as a modeler, we think. And it is very important that we focus on our and model it accordingly. So first I will talk about some historical challenges, background. I will also talk about epidemiological aspects in COVID-19. And then I will talk about some of my studies on modeling COVID-19 aspects, eventually ending it with future modeling challenges and limitations. So, the pandemics have nothing been new to us. Maybe in my lifespan, there has been now. Prasad, can you hear me properly, or is it from time to time my connection is not very good?
We can hear you, sir.
Okay, fantastic. There has been many pandemics in the past, and we have learned quite a lot by analyzing data and using models, what has been successful and what has not been successful in the past. For example, in 1918 Spanish flu, we did not know even about the virus, that it is spread by influenza virus. It was spreading by influenza virus. It is usually people used to think it is maybe a bacterial infection. It was not until 1933 that that the virus, human influenza A virus, was isolated in humans. So it was very late. One of the key features that recently was published almost 2 decades ago, or less than 2 decades, was published about using data from 2 different cities, which was one was Philadelphia and the other one was St. Louis. You see on the top right of my slide, there is a 2 graph of Philadelphia and St. Louis. Philadelphia has a very high peak and St. Louis had a very low peak and spread out outbreak. And one of the reasons that the people that analyzed the data for these 2 cities recognized that the reason that Philadelphia has a high and a bad outbreak in some sense, because they waited 2 weeks after their first case to implement the social distancing policies, whereas St. Louis implemented it in 2 days. So that resulted in something we have been hearing quite a lot in the news. It's called as flattening the curve. And the flattening the curve is on the right. You are seeing if you implement certain control measures, then basically the peak comes down and the curves kind of flatten up. And well, but there is an issue here that we can't just implement the social distancing policy and flatten the curve, because there is something called as a hospital capacity. Because we don't want to overwhelm our hospital capacity, and that is a horizontal line. And you see the 2 horizontal dotted lines are there. One is the initial before an outbreak, and slowly you want to raise that health capacity of the hospitals so that it can handle more cases. For example, in New York, they had this big area conference venue that they converted into hospitals. So they increased the number of beds for the patients of COVID. So this line is actually not stable. This is fluctuating line. So there's uncertainty associated with it. And this keep on changing on a daily basis, and beds are getting added. There is a lot of variation in these type of things from city to city. Well, the flattening of curve has now you don't have to, if you have left your health capacity to a previous to COVID level, you may have to do a lot more social distancing protective measures. Then slowly, when you increment your health capacity, you may relax social distancing and maybe open up the cities, for examples. So these are these variations. Usually modelers, as a modelers, we have to think about not only in terms of a disease outbreak, how many cases, what is the peak and all those things, but also as a level of healthcare capacity that could handle number of cases that are occurring for the COVID outbreak. So this is the timeline for the diseases, for the COVID outbreak, starting from early January. In fact, in China, it was in December, and some says even in November, there was a first case reported. But we started to see the big news about COVID-19 starting January. And well, as a modeler, what is important is that uncertainty in the information has changed. For example, initially, we only know a little bit about epidemics characteristics, whether it is spreading in clusters or whether it is spreading everywhere, or whether there is asymptomatic cases are spreading it, and so on. So this information slowly started to become more widespread, and therefore the uncertainty in the epidemic information was reducing. But then there was other information that was coming as far as policies are concerned over a period of time that we started to see uncertainty in. So the evolution of information that we use in the models have changed drastically from being more less precise to more precise now than before. And there are still many things we don't know about. And if we model things using those information, we may be limited by our capacity of modeling methods to draw a reasonable conclusion. So this slide is about evolution of models and evolution of information that is used in the mathematical models, for example. Well, I came across this BBC interesting article, actually interesting in some sense, but it's sad in other sense. And it was on, and I kind of thought over this thing quite a lot. This was written by a journalist. She's an Indian, and one of her family member died in Gujarat fighting with COVID. And well, she writes about how devastating things have been because our system is overwhelmed, because there is a fear in the systems. And these things are very critical for the models to understand, because human behavior plays a very important role in the spread of the diseases. So what she says in this article is, so as the infection was, as her relative was, his condition was getting bad, she was doing many things, and she was a BBC journalist, so she's very well known. In fact, she knew many people, in fact, many of the political leaders and other journalists, local journalists in India. So she said, I called hospital after hospital. I was told there was no room, not a single bed in the entire city. Repeatedly calling helpline, got nowhere. Finally, after being admitted one day of his ICU, intensive care unit, I was able to see him struggling for oxygen in a video call with me. A strong man who was exercised daily and had no health issues. He could never have thought that a virus would land him in this situation. Well, it is really painful reading this article, but really an eye-opener that there are hospital settings or healthcare system and it's not about India, it's about whole world I'm talking about. They are being overwhelmed. Usually in developing countries like India, they have been overcrowded, understaffed, long wait hours because resources are limited. It's a developing countries, not a developed country. Even in developed country like US, in New York, in spite of being the best city, has to face many of these challenges. So there was no ventilators, for example, there was no good communication. So now the question comes, what as a modelers we can do for that? I mean, of course, we read these stories every day and we become sad and we kind of post these things on our social media accounts. But what exactly we as our strength is a mathematical modeler. So what can we do basically? So, well, there is lots of models that are already established by mathematical modelers in the past. We can use many of those things. So for example, queuing theory based models have been used for management of hospitals, for example. So if you see top left corner, there's a picture where there is circles and circles donate the hospital settings. So this person who is as a patient is coming in to some centralized hotline and asking about where are the beds available. You need to we need to streamline it effectively that they don't die on waiting to get into line for getting a bed, for examples. And as happened in this case in BBC article, for example. So the queuing theory has been amazingly helpful. The models using queuing theory here, we talk, there is certain inputs that goes in such type of model and it optimizes where an individual should go, which hospital individual should go, and where is the availability, rather than randomly going to the hospitals and wasting time and basically making risk for the life of a patient, basically. So just to give you an idea, this model has input like arrival process. For example, how many patients arrive in a queue in a given hour? What is an average time interval between arrivals? Does that vary? The arrival of the people coming to the hospital depends on a time of the day, whether it is morning, lot of patient coming of the COVID, whether evening is. Queuing process, how much time patient is waiting. Suppose you are in the hospital, that is a best availability that you can get. How much time you have to wait there to get the bed finally. So these kind of service, for example, what would be if the hospital is overcrowded? How do you decide first come first? How do you decide who will get the first bed, for example? So there are lots of these inputs that goes in the models, and it helps you answer certain output of the model that I have it in my bottom right. For example, it helps you optimize the results over average weight per patients. How can you reduce average weight patients? What percent of time that a healthcare employee or a machine that is used on a patient are being utilized? You don't want to waste the time just by sitting around where other places person is dying because there is no machine, for example, or there is no people to healthcare worker peoples. Here also you can talk about capacity, raw materials like PPE, protective gears, for example, or ventilators. How many you need in a certain location? So these things can be done very interesting using a queuing theory based models. And behind the scene, they are using something called as a little law, which describes the relationship between number of patients in a system and a mean arrival rate, and the length of patients remains in the system. So how much duration the patient stay in the hospital, for example. So very beautiful modeling techniques have been used in the past but it seems like in India we are not using it, and in other countries also these modeling techniques we are not using, that we can use it, for example. Okay. Well, another story that I came across in a scroll.in, it's an Indian story that talks about there was in BHU, there's a diagnostic lab. There's a Institute of Medical Sciences. That's a Department of Institute of Medical Sciences in Banaras Hindu University. And there is a diagnostic lab that is handling 30 million people. And for just one test lab is testing for 30 million people. Think about that. That's a huge number of people. It's one lab in Eastern UP, Uttar Pradesh. So it seems like we are understaffed. The testing facilities are very limited, well, in this case, they are getting samples every day. 1,000 samples they can handle in 48 hours. They take 48 hours also, and they are getting around 1,700 samples a day. So. Well, huge amount of samples they are getting. So they are prone to make an error into it. Because they are overwhelmed with the work and they are dealing with a lot more people, a lot more region. These tests will be prone to lot of error. So how do you model those errors, for example, and what those errors will lead to? How many new cases? What changes it will do to the outbreaks? So I mean. There's a models for imperfect testing on the disease dynamics. You can talk about imperfect testing. Now, in this picture that you are seeing here, there is one patient that we are following, and these colored lines indicate different types of test. Well, there is antibodies test, there is antigen test, there is actual virus detection test. So it depends on what you are testing. So PCR is a virus detection test. Polymerase chain reaction is the test. That basically identify a live virus in the body, in the specimen or sample. The only time you can use it if the live virus is a lot in the body. But as the progression or initially you may not find the actual live virus because it is very small amount of virus in the body, and the test will say no, whereas the test may make a mistake there in identifying. Or later on also, after a person has recovered, if you do the testing using PCR, you may not be able to find the virus, and it will give an error. It will say the virus is not there, whereas the virus is there, but at a very low level, right? Then there is another test called as antibody test, where antibodies are getting. Antibody test you cannot find in the initial stage of the infection because antibodies takes little time to develop in the body. So this test starts to fail if you do it very early on. Antibody test will not give a correct result. So the question is, how do you model? And how, if you model it, how do you understand the disease dynamics on it, basically? So, well, before I go into the actual model component of imperfect testing on the disease dynamics using these two tests, PCR test and antibody test, we need to understand what is the main, how do we say that the test is creating a bad result, for example. So for example, test, so if you see this table is showing test being positive and test being negative. If a person is infected, if the person is not infected on the columns. So there were total infected, their total number of people in the population was N. Out of N, A plus C were infected and B plus D were not infected. Okay, so A plus C plus B plus D is equal to N. That's the total population. Okay, so the question here is, if you apply a test, you can fail to detect or you identify it correctly. So there are 4 different choices. The test is saying positive and the person is positive. That means person is infected. Or the test is positive, but person is not infected. Or test is negative. That means test is saying not infected, but actually the person is infected. Test is saying not infective, but the person is also not infected. So in 4 of these values, 2 times you made an error and 2 times you made a basically correct decision. So test sensitivity, any test has something called as test sensitivity and test specificity. Basically, it captures how many test sensitivity captures how many correctly you have identified among all the people who are infected. Okay, and test specificity is how many correctly you have identified among those without the disease. Okay, so this is A and D and divided by total population in that column, by the way. So these are the 2 parameters that people use for identifying how bad is the test or how good is the test, basically. Okay, so there is simple model we can use for it, is SIR model, which is susceptible, infected, recovered, which is a simple epidemic. And here you can build in virus active tests in infection test. So there are 2 possible tests: active virus infection test, which is a PCR test, and then antibody test, which is basically testing for antibody. And here the testing rate is basically given by sigma A and tau A for active virus, and antibodies you are testing it by sigma B and sigma tau B. And basically it depends on antibodies test. You will not do it in a current infection. You will do it in a quarantine people who are already recovered from the infection. There you want to see if you have an antibodies or not. Whereas active infection test, you will do it in a general population because you don't know if they have so you will do in unquarantined individuals. By the way, QS, QI, QR are quarantined individuals here. And you can build in sensitivity and specificity in the simple models, and you can study what happens if the error made by the test varies. And error could vary because of many different reasons. Because of the test, the raw materials of the test are bad, or test handles in a very incorrect way. So these things we can study using a very simple model like this one, and study what happens over time if the test efficacy we know is this much. Okay. And that was happening in the BHU, Banaras Hindu University Diagnostic Test Lab. They were making errors, but we don't know how much of that error is causing the outbreak to grow or reduce, and over what period of time. And such kind of models can be used very easily to understand this thing. Now, again, I'm giving you a broader perspective about the modelings of various relevant features in the community with the proper examples that we are seeing currently, rather than just making up something here. Okay, so this is a very good example and very related to the examples that I gave from the Banaras Hindu University Diagnostic Lab. Now, well, another aspects in the modeling that we face is about definitions. Well, I think some of you might be aware of, but this is something came in May 22 news that almost 40%, 35%, 40% cases are being don't have a symptoms. And if they don't have a symptoms, how will you know that they are spreading the case? So they are. What is the name we should use for that? Whether we should use latent, whether you use asymptomatic, whether you use pre-symptomatic. What is the name? And each of these words has a precise definition in the literature. Okay, so for example, asymptomatic means people who are not showing symptom, but will never show the symptoms in future also. They will recover without showing symptoms. In previous diseases, we might use this kind of terminology in a different sense, but here asymptomatic is used like a self-healing, where you don't see the symptom at all. And pre-symptomatic are the individuals who are right now asymptomatic, but eventually will going to show the symptoms. And these two folks, these two groups are causing lots of infection in the communities, especially in the COVID case. So definitions have to be properly modeled in our mathematical modeling equation before actually we talk about mathematical analysis, numerical simulation, linking with the data, and so on and so forth. So this is one thing, and I think if there will be lots of cases which will not be showing symptoms and spreading the disease, then the best thing you can do is either not go out at all out of your home, social distancing, or wear the face mask. So one could even evaluate the impact of these measures more accurately if you define the terminology in a proper way. So we'll talk about some modeling methods now, and some of you might not have a background in modeling methods for pandemics. So let's talk about for an outbreak. There are two basic questions that we talk about: contagiousness, that means how fast the disease is spreading in the population, and you might have heard about the number which is called reproduction number, that is very common in the news articles nowadays. And then the second one is how virulent or how dangerous is the disease. That means how many people dying from the disease. And these are captured by case fatality ratio or mortality rate of some sort. In this table, you see there are different diseases with different reproduction number. Measles has a high reproduction number, but not medium case fatality ratio, whereas MERS had a very high case fatality ratio, but had a very low reproduction number. Low reproduction number, how it is spreading, and case fatality ratio is how deadly it is, the disease, basically. Okay, Well, this case fatality ratio is why I'm talking about these two metrics. Let me go back actually, why I'm talking about these things. Well, this is relevant when we talk about a disease like COVID, these two outcomes of your models. If we do not provide these outcomes from your models, then we are giving only partial information about an outbreak. These are critical informations. And besides that, you can do many other things. But I think some of these aspects needs to be incorporated very much in your as an output variables from your models, basically. So case fatality is basically a ratio of how many people died divided by how many diagnosed with the disease. Okay. Now, this is a disease. So how many diagnosed with the disease depends on how many you test, basically. Some countries are testing quite a lot. Some are not testing very lot. For example, South Korea is testing a lot. And so therefore, their case fatality ratio will be very low. Whereas in US, India, and many other countries, it is not as high as in South Korea, for example. So that depends on the denominator of the case fatality ratio, which is number of people who are diagnosed. So this metric may not be the best one unless you can do a actual sampling study where you can follow the people and then you can estimate case fatality ratio in the cohort of the samples. So it depends on CFR, depends on surveillance system, testing capability, testing efficacy, but also it depends on who we are testing. So for example, we may not be able to identify asymptomatic and pre-symptomatic cases, which are not showing symptoms. So therefore the denominator will decrease. It could also depend on age. For example, some countries, old people are dying quite a lot, whereas some other countries, some other age groups are more dominant. So, so age structure matters quite a lot when you are calculating case fatality ratio. So this number is kind of delicate and has to be used in a very cautiously for the output variable. So I'm showing in this picture, right corner, how is the case fatality ratio is changing over time. Why? Because you are testing now more and more. So as the time is progressing, this is in China, in the 4 different places in China: Wuhan, outside of Wuhan, outside of Hubei. Hubei is a province or a state of Wuhan city, and China overall. So this has changed over a period of time because they were doing more and more testing, and therefore denominator is increasing quite a lot. So this numbers is showing inconsistency in the output. But again, it is the best thing that could measure the fatality. Some people also use crude mortality rate. In the crude mortality rate, the denominator is the total population, not the total cases diagnosed, but the total population. And sometimes people use that one to draw a conclusion. But then the number of deaths could be highly underreported also. Well, as a modeler, we ask ourselves many things. As the outbreak is going on, we initially ask ourselves, how do we prevent? If there are few number of cases, how do we prevent? Like, for example, SARS outbreak, or not SARS, sorry. Yes, SARS also. SARS didn't become a pandemic like COVID. So it spread, but we were able to prevent its invasion or reinvasion due to many social distancing measures. SARS outbreak was in 2002, 2003. What is the growth rate of it? How fast it is increasing? The outbreak is increasing. It is very important metrics, and we used many different models for measuring how fast it is increasing. Okay, this is very, very critical, and this could be not necessarily for the whole world or for the whole country, but you can talk about your city or your neighborhood, how fast it is growing. And it depends on the local structure of the community. What interventions would be helpful for the population in hand? How drastic intervention we should implement? These all questions are modeled and are captured and asked by as a modelers. So we do it all the time answering this thing, and we are doing it for COVID-19 as well. Well, before I go into an examples from my research, I need to give some basic background on the this disease models. To some folks may not have an experience in that. So disease model, basically what you do is you consider whatever population that you are trying to study the disease in, you divide that population into three compartments, basic three compartments. Susceptibles, people who are not infected, infectious, people who are infected and infecting others, I compartment, and R, people who have recovered from the disease. And then you have the arrow coming from susceptible to I to R. And that is called as a transmission. That means new cases. Transmission is the new cases that are converting from S to I due to infection transmission, and recovery, that is cases are going from I to R. Now these two arrows are modeled in a very, very different way. What if people are changing their behavior? So transmission rate will decrease or increase based on how strictly one is following, people are following social distancing, for example. But in general, these two I compartment, if I look, it is a function of something coming in and something going out. And that something coming in and something going out decides the level of I compartments. And you see, if we simulate it, the I compartment will look like a red curve in this picture. Okay. So that's a very simple thing. Now, these transmission rate could be a function of host population size, contact rates, how fast they are moving, pathogen infectivity, like how bad is the pathogen in transmission itself. So again, there is a quantity, we call it as a reproduction number, average number of new cases that we talked about a few slides back. It's basically number of new secondary cases each infected person is generating in the population. Now, this is an average number of all, average it out over all the infected people in the population. But this number is beautiful because it tells you few things. It tells you first, it tells you the ratio of how much is coming in and how much is going out in the I compartment. So this is beta over gamma, how much comes in and how much goes out. So if beta over gamma is greater than 1, that means more coming in and less going out, then there will be a peak. That means R0 will be greater than 1 and there will be a peak. If beta is less than gamma, then there won't be a peak. And you see the top two graphs are showing R0 greater than 1 and R0 less than 1. In the first graph, there is an outbreak or there is an epidemic. And in the second graph, there is no epidemic. Okay. So this is an epidemic threshold. That's why we call it as an epidemic threshold, because it tells you whether there is an outbreak or not. It also tells you how quickly you will rise to the peak. So if you see in this graph on the third graph, that higher the value of R0, the quickly it is rising to the peak. There are different values of R0 that this picture shows, and it shows the peak is high and very early on. So it tells you how fast the epidemic is growing also. It also tells you what will be the size of the peak, how many people total will be infected, and it will also tells you what level of immunity or what level of control programs we will do in the population to control the disease. So there are many features of this reproduction numbers that we use in defining an outbreak. Well, just to give you an idea here, these are 4 diseases that I'm comparing: measles, influenza, chickenpox, rubella. And measles has a very high reproduction number, and influenza has moderately high, but chickenpox is much higher than influenza, and rubella is lower than chickenpox but higher than influenza. So you see these graphs that we are seeing on the left side, the peaks, and if these type outbreak of these things will come into population, it will show the peaks like this, assuming everything else the same, basically. Now you can make this type of modeling more complex. For example, recent news that I just heard is that you may not be having a permanent immunity of the COVID. You may recover a few time, a small period of time, and then you become susceptible to disease. You can get the disease, and in that case, it is not simply SEI model, or E stands for hidden infection, asymptomatic, let's say. But the second graph would be more appropriate if you are talking about that you may get the infection again. In that case, the recovered compartment would come back to the susceptible compartment. Okay. Prasad, can you let me know 10 minutes if I'm before my end period of 10 minutes? Yeah.
Oh, okay, Anuj, I'll do that.
Sure.
I think you are now till 20 minutes is there.
Okay. Thank you. Thank you.
Yeah.
So in this models, you can make it very complex. For example, you can incorporate T compartment, which is a treatment compartment. Although for COVID, there is no treatment, but the treatment also means providing ventilators, for example. Treatment doesn't mean only drugs here. So treatment is therapy, some kind of therapy that you are providing. So how do we model? Which models we should use? Of course, it depends on a research question in hand, but it also varies upon what shape we are trying to imitate. So if you see on the bottom right corner, it is showing a different shape based on what you incorporate in the model. So what we are seeing right now in the COVID is a third situation, which is we are expecting the second wave in some countries, by the way. In India, it is still a first phase, but second countries, there are countries which are expecting the second phase right now. So it could be the last figure. That would be a better to model that way rather than first two figure, for example. Well, as a modeler, what do we do? I mean, I think some of us do these things so regularly, but we never discuss. We never discuss about it. There is lots of uncertainty in our task, basically. So precisely what we do is these 5 things as a modelers. First is identifying the sources of the mechanism. That means we have to know the research questions. We have to know what are the factors that are driving the research questions, and that may be critical to the dynamics. So 30% of our work time efforts goes in the just identifying those sources. And then incorporating those sources in the mathematical models, 10% roughly. That's very, very small amount as compared to identifying the source of mechanisms. Well, performing the mathematical analysis and statistics is also, these are not absolute numbers. These are variable number, but approximately 20% of the effort goes in mathematical analysis because we are used to doing these mathematical analysis. And then interpreting the results. Interpreting the results takes a huge deals of information and connecting it back to the first point, which is mechanisms. And that takes 50% of and eventually we need to validate the results using an alternative source of who says that our results are correct. You have to find a ways to identify how do you validate these things. So these process we do it now and then, but the amount of percentage, according to me, it should be like this distribution. The 30% in the top one and the 25% in the bottom one are the most important time that we are spending in understanding our mathematical models. There are different reproduction numbers that we talk about in the literature. There is lots of uncertain aspects. There is confusions because people use left and right reproduction numbers, but the meaning of it is very, very different. So the basic reproduction numbers that I talked about is only at the starting of an epidemic or starting of an outbreak. Effective reproduction numbers is a time dependent and you can talk about any time. Control reproduction number only you talk about when the interventions or the controls or some kind of like face mask, social distancing is already there in the population, then you talk about control reproduction numbers. and type reproduction numbers when you might have a different type of people, right? For examples, you could have age group. So you can talk about old type reproduction numbers versus young type. So how many old people are infecting and how many young people are infecting, for example. So that is called as a type reproduction number. Now, invasion reproduction number is also there. That is when there is multiple strains. So suppose COVID pathogen strain mutate and there are maybe 2-3 strains of it circulates in the population. In that case, you will model those multiple strains. In dengue, for example, there are 4 strains and people model it. There you talk about when one strain is there, what is the reproduction number of other strains to invade. So there are many of these reproduction numbers. That there are other reproduction numbers, but these are important ones, and so I listed it here. And so there should not be a confusion. We should always ask her, people use reproduction number, but which one you are talking about? And it will, the meaning will change and the measurement of that will change as well. Now lately we have been hearing something like a herd immunity, and one of the examples of the COVID comes into mind is Sweden. Sweden is actually doing something called as they are trying to achieve a herd immunity, and that's why they didn't do any kind of lockdown, and they let people do everything except they said, okay, you have to be careful, don't be too close. It was really in informal ways that they are kind of pressing forward to that. Well, a recent study. So if there is a question.
No,
Yes.
we can continue. And there are 10 more minutes are left for you.
Okay. Study showed 6.6% Sweden population has antibodies only till late May. And actually 60% is assumed to be herd immunity. Herd immunity means that you should have sufficient number of immune people in the population that infected person, when they go out, they don't get enough of people, susceptible people around them to infect others. And so some people are providing immunity to other people, and that is what is called as a herd immunity. So it is believed that 60% people have to be immune in order to be herd immunity. Well, it is calculated in a very, very simple way. It is calculated using R effective reproduction number. And if the effective reproduction number is less than 1, that means you are not growing, the cases are not growing. Each case is not replacing him or herself, basically. So it is R effective or effective reproduction number is R0, basic reproduction number, times the ratio, how many people are susceptible, fraction of people who are susceptible. And if this number is less than 1, then you find out proportion of susceptibles that needs to be, if you make this thing less than 1, you can compute something called as herd immunity. Very simple idea. And we are talking very fancy way, but it is computed in a very, very simple way, how herd immunity in this thing. So anyway, and as soon as the herd immunity goes up, R effective number goes up, there is a second wave, there is a risk of second wave coming back, basically. So let me talk about some contact tracing modeling. The question is, we don't have a treatment, we don't have a vaccination. So what can we do? Well, contact tracing is one of the best methods. But there is a meaning for it. The people use word quarantining when they are basically isolation is another opposite of quarantine. Both way you are restricted some way. Quarantine is more voluntary, whereas isolation is forceful, that you are asked to remain inside. And basically what you do is, if you are infected, you basically, if they identify the person is infected, they trace the contacts. So how do you model these things? Basically, you can model something like that. If there is a model like SEIR, and basically forced is an isolation, Q3 is an isolation, and Q1 and Q2 is quarantine, because you don't know in Q1 and Q2 whether they have a symptoms or not. They don't, from their face, they don't have a symptoms. Whereas I is showing the symptoms, and therefore you are isolating it forcefully. Okay, so so Q1, Q2 is quarantine and Q3 is isolation here. And you can model this thing very beautifully, contact tracing, even though contact tracing is an individual behavior, but you can model it. So for example, contact tracing you can say is fixed efficiency. So if you see my second point bullet here, which is basically capturing how many people from S to Q1 you are taking out and E to Q2 you are taking out, there is K times you are doing more for susceptibles than for exposed class. So that means you are somehow quarantining K people more. So if K is equal to 0, perfect efficiency of tracing. That means everyone who's infected, you are isolating only those, or you are quarantining only the cases which are infected. But people, but if K is equal to 1, K is equal to big number, maybe 50 or 100, then you are basically wasting your efficiency resources. Efficiency is not very good for contact tracing. So beautifully it is done in a very simple way, how the contact tracing could be done. But anyway, I mean, you can talk about cost here. You can model cost associated with it, different types of cost, public health cost, societal cost, direct cost, indirect cost, cost to an individual. We can model all these things very, very cheaply. And there are methods to do that. And this is one of my paper in the past where we model cost associated with quarantining and isolations. When the contact tracing efficiency is there, basically. Well, the result is there. What is the training efficiency and how much will be the cost? It is not always more contact tracing, always the better. There is a threshold in that, and that would be needed for controlling the disease, which will be cheaper as well as it will control the disease. Well, there is another studies. Prasad, I have 5 minutes, or I'm out of
5
minutes.
5 minutes.
Okay, thank you. I will be done with that. Promise. Well, you know, recently there was a news actually about Delta Airlines changing their policy of boardings. In US, there are planes. They are boarded. People aboard the planes by zone wise, and they take zone 1, zone 2, zone 3, zone 1, zone 2. They have decided it based on optimizing their time. The time is the money for the airlines. So they have done it based on their making so fast so that people can go fast and therefore we can save money. I mean, the airlines can save money. But that policy may not be true for an outbreak if the outbreak is spreading, for example. So recently Delta changed it. In fact, Delta, American, many airlines changed it from back to front. That means back row comes first and then the next row from the back, and then the first row in the very end, basically. So the question is, instead of doing the zone wise, which used to be the case, now they are doing is back to front row wise. Is this a good policy, or they just made it a random decision? Well, it turns out, by the way, it spreads in the, it is very much likely to spread in because it is airborne and asymptomatic can spread it, basically. So it is very much through the air travel. So this is the question that we had, whether their policies are better or not in the plane. So we developed these models where basically which we are capturing is probability. These are individuals in the plane. Think about people who are entering the plane and they are susceptible. There are some infective and they are coming in close contact with each other. And the contacts here defined as the radius of infection as well as duration of the contacts, how long they are next to you, basically. And basically you can model something like that. And we did some of these things with a huge simulations, and we found out the policies that Delta and American Airlines are doing are actually counterintuitive. They are causing more harm than good. In fact, it is better for them to go back to the previous policy. And this paper is actually in the review right now, but it causes lots of uncertainty in that. And And this is the way the modeling has been extremely helpful. So with that, let me stop here. Thank you very much for listening to me, and I hope I have motivated enough to provide some basic idea about how we do the modeling, what aspects are critical, how do we relate it to the real life aspects of it rather than just a theoretical aspects of it. So thank you.
Thank you, Anuj. There are few questions from the participants. Yeah, hope you are able to hear me.
Yes, I can, loud and clear.
There is somebody is asking that how China recovered so fast.
How what? Say
how China recovered so fast.
How China recovered so fast? Well, it will be too early to say recovered, because China has an outbreak coming. Reporting. I mean, I just read in the newspaper that that there will be this Beijing is infected, is reporting very high number of cases, by the way, and they are closing down all the cities and they are doing the similar type of intervention as Wuhan, by the way. So I don't think that it is recovered, but as I said before, they might have crossed the first wave. The second wave is going on right now in China, by the way. It's starting to.
Okay. There is one more question from one more person. Is saying that some of your models show that there is permanent immunity in COVID-19 case. Is it so?
Yeah, so if you see, my title was Uncertainty and Precision. So, well, we did not, up till recently, we did not know that is it a permanent recovery or is it a temporary recovery? Well, we still don't know very clearly whether temporary recovery is the right way to go forward with the COVID or not. But as far as the past experience on a coronavirus and influenza strain is concerned, for that particular strain, you could, you might have, once you get it, you get permanent immunity. So again, this is what my title was: Uncertainty and Precision. There are so many uncertainty. Maybe in months to come, there will be more studies where we will make a more precise guess, a more precise answer that, okay, there is no permanent immunity, for example, in that, and then we will change our models. But right now, this is the best knowledge we have.
Okay. There is two more questions, and one is some of your models doesn't include SCR type models, only SAR like that only. Why it is so?
Well, the models that I showed were for SIR was just to show, just to build up the theory there. And well, you are absolutely correct that, as I said, the definition of asymptomatic and pre-symptomatic is very, very important. And most of my models right now has been having those type of things, although I didn't showed all this, all the studies, but in the plane also. I wanted to give an idea about how we are modeling it rather than exactly what we are modeling. We do have asymptomatic and pre-symptomatic stages. But again, when we are talking about airplane, for example, the duration is 2 hours. It's not the community that we are talking about. In community, people live for good number of days, basically. But in flight, you stay there for a few hours. So here, the differentiation of asymptomatic, pre-symptomatic really doesn't matter too much. Okay, so it depends on what is my research questions that we could focus our attention to. Yeah.
Okay, this is question from my side.
Sure.
How to validate the models of pandemics such as COVID-19? We have a lot of different data is coming from our side.
Yeah. Well, there are, usually in the field, people use it half the data. If you have 100% data, you use half the data for estimating parameter and another half for validating your work. Okay, so now what will be your half in this case? Well, suppose you have incidence data, like number of new cases that are coming to the hospitals, time series data, that you can use it for estimating parameter. And then you can use a mortality incidence, how many people dying, to estimate, to validate your model, whether that is making sense or not. So usually you have to have some alternative. Well, right now people are also using cell phone data. So how people are moving, how people are following a social distancing, for example. So these apps, we use all these apps, and these apps, once we use it, they report to some collection agency, and we have these data that we are using to identify how people are moving in social distancing, for example. So those type of data also we could use it, right? So there are different data sets that one could use it, one has to use in order to value. You cannot use the same one, basically, as you are using it for parameter estimation.
I think two more quick questions. Somebody is asking that is it possible to share your PPT?
I think it is a recording you will going to post. Or?
Yeah, recording I'll post it.
Yeah, so they will have all these things.
Okay. And one more quick question: how to manage the economical aspects with the mathematical modeling during an outbreak?
Well, I showed one example of taking into see when we say economic, what do we mean by that? I mean, economic for who? Well, we cannot do everything. Basically, it's a human mind can understand only limited information. The reality is very complex. And so we need to be focused on, for example, I showed in my slide different types of cost to the individual, cost to the public health, cost to the society, right? Productivity cost. Productivity is if I am sick, I don't go to the work. That is a cost. There is a money dollars amount that I am not earning, for example. So that is a cost. So what we are measuring it, first of all. And again, the same way that I showed, I mean, you can find out average 1% cost of doing something and then multiplied using your epidemic model, how many people are showing those type of things, and then you multiply that unit price with the total number of people who are infected. So that will give you a total cost, for example. So yeah, I think Jean Marie Farrell also has a question. I saw her hand raised.
Yes. So thank you. I was just waiting for all the mathematical related questions to be asked in Jen, first of all. And thank you so much for the wonderful presentation. And can you share any case studies in which the mathematical modeling was successful so far in any of the viral infections? Was there any case studies on this? Because we normally don't see the mathematical modeling when we are working on chemical experiments. So can you share with us any success stories of this mathematical modeling and how far it was useful in the past?
Yeah, well, there are many actually, not one or two. There are many studies that has been successful. Well, in HIV, for example, the whole field has been very successful because of mathematical modeling tools, because HIV is a long-term disease and there is hidden, people do not come, social stigma is attached to it and people don't come forward to it. There's many ways, for example, when we run this thing, how many drug resistance cases will be there? So so, you know, they use these treatment for HIV for pre-exposure prophylaxis, which is like a vaccine for HIV, which is given to the vulnerable population. And well, if you give a lot of treatment in the population, how do you trade off? I mean, the benefit is you are giving more treatment in the population, but then you are causing more drug resistance cases also because you are pressuring the population with lots of treatment. So there is a trade-off. What is that threshold? And we have seen it in some population who has not built a models. Their cases has been huge as compared to some places, and we have done studies in South Africa, for examples. In COVID also. Now, one thing I want to, this is actually a very good question you asked. One thing I want to say here is, if we are talking about projection, okay, how many cases will be tomorrow or day after tomorrow or 10, I think there is not many studies. But if you are trying to understand which group I should, where should I find the clusters of the cases? In Delhi, they are identifying clusters using models. Also because what happens is the case might have reported in this neighborhood, but got infected somewhere else. So this may not be the high risk area. And if you do just by clinical study, you may identify this as a high risk area, but the high risk is this area where the person got infected. Okay, so these things, identification of the right clusters, in Delhi, they are doing something like that using mathematical models, for examples, and I think other areas also there people have done it in a how the system work is the role of a mathematical, not really by as a projection, not necessarily how many cases will be there or how many deaths will be there. That is weaker things to project because there's so many uncertainty. But you can understand what drives the system, what aspects of the models are changing the system's behavior, basically. That you can understand.
Okay, thank you so much, sir. I think this is a more informative presentation by you. And I would like to address the question what you have asked me in the beginning. And I think first time, I mean, we are in contact and you are in VIT, I mean, virtually. And I would like to extend my invitation to you to come to the campus and you can visit us and you can join with the speakers, I mean, the organizers here who is known to you. So we would like to have collaborations with you and maybe it can be a joint proposals or joint publications and also you can be the visiting professors. So we will be very much happy to have you. And my invitation is to all the speakers present here. So I'm welcoming all of you to visit campus when the situation is becoming all right. And probably after September or October, I think mathematics department is planning for one big conference, which is in association with Mathematical Society of India. So if everything is getting all right, we may be having you in person. Otherwise, again, looking forward to meet you in virtual mode in the month of December. So thank you all. And once again, I would like to thank the speakers. So
well, let me say one thing actually very quickly. Sorry, Prasad. You know, I showed an example of this airplane models where the boarding policy, we are suggesting a boarding policy which is very good for it. And that's a very good example of what the question you are asking. That is practically being used right now in decision-making policy, for example. So I think the reason we are making it so that we can quickly come to the VIT conference and give some talks there as well and meet folks. So thank you for your invitation. Thank you.
Thank you, Anuj. Yeah. Bye.
Yeah.
So I will drop right now because it's too late in the night.
Yes. Thank you, Anuj. Thank you for your time. I know it's very late night for you. Thank you for your invitation. And there are a few more questions. Maybe I'll send you a mail and we can get back to you and I can contact them, the answers, I can send them back.
Sure. It's my pleasure. And thank you for inviting me. All of you, it's very, very kind of you all to invite me for this. It's an honor. Thank you.
Yeah, thank you.
Bye.
Okay. So, Ashish sir, I'm sending you live. You can yeah, you are online, sir. Ashish sir.
So
sir, Datti sir, you are in mute, sir. Datti sir, can you unmute yourself once?
So our next speaker is Professor P.S. Datti. Professor P.S. Datti did his master's from Karnataka University, Vellore, and joined prestigious TIFR as a student, where he was deputed to Crude Institute of Mathematical Sciences, New York University, New York, to pursue higher studies. He did his MS and PhD from Crude Institute of Mathematical Sciences, New York. While pursuing for PhD at the Crude Institute, he taught undergraduate courses in New York University, Baruch College, and Cooper Union College. He has written notes for TIFR lectures with Professor G. Vijay and Professor Kathleen S. Moravec. He has co-authored two books with Professor A.K. Nandkumaran, the first one being Ordinary Differential Equations, Principle and Applications, and second one, which is yet to come, is Partial Differential Equations, Classical Theory with Modern Touch. And both of these books are from Cambridge University Press. So I request Professor P.S. Datti to just continue his talk and just explain how we can fight with COVID-19 with the help of mathematical model and how mathematical model is effective. So, Datti sir, please.
Sir, Datti sir, can you share your screen, sir?
Yeah, sure. Is that okay?
Not shared, sir. Screen is not shared.
Oh, okay. I just.
You can click on the shared screen.
Okay.
Yeah. Yes.
It's okay, right?
Yeah.
Shall I start?
Yes, sir.
Okay. So, thank you very much for the introduction. And once physically visited VIT, so it's again my pleasure to be part with this webinar. And am I audible? Okay.
Yes, sir. You're audible.
Yes, sir. You are audible.
Thank you very much for the invitation to this webinar, and I thank Ashish Kumar Prasad for this initiative and his colleagues in the math department for taking this initiative to conduct this webinar. So because of this lockdown, we have lost many things, but slowly we are coming back to life once again, including academic activities. And this is really a good occasion to share some of the mathematical ideas in modeling this diseases, especially in epidemiology. Let me just say one thing before starting. So I am a differential man, and of course, as we know, all the differential equations are phenomena in science and engineering, including, of course, biology. So here I'll be talking of some models using ordinary differential equations. Okay. So let me begin my talk, and so I'll begin with some general remarks and then discuss two models. So it was really a nice informative talk by Professor Anuj Mubayi, just now we heard, and he has explained so many biological words which I don't know. I am learning, and I am sure many in mathematical community are new to these terminologies. So it's very important to keep those terminology in understanding what goes into this model. So for example, SIR model he briefly mentioned, so I'll give more details, and I have my own questions regarding these mathematical models, especially in the wake of COVID-19, and I'm sure there are many still unanswered questions, how to go about modeling itself for this COVID-19, because we are seeing every day a new and newer phenomena, and also the local governments are taking different strategies depending on the spread of the disease. So let me start with some general remarks. So communicable diseases such as measles, influenza, and TB are a fact of life. So we have to live with them. So especially, for example, TB in India, before this COVID-19 came into picture. So this TB is still prevalent in India very much. And there are many more mathematical models in understanding that spread of that disease and why it is still present in the population. Okay, so these are just some terminology. Epidemics are sudden outbreaks of a disease, just like this COVID-19 we are seeing. Earlier also, the populations world over faced so many epidemics, and some are recorded. So, and so these endemics are the situations where a disease is always present, but hopefully there is a medicine for that, and like TB, influenza, and measles. So it's elaborated on so many things, especially in public health domain and other things. So here I have listed some of the questions the public health personnel and in general the local governments will ask, and these are the so how severe will be the epidemic? Everyone wants to know, even why only public health personnel or governments, but even individuals, and how long it will last, how many individuals be affected and require treatment, what is the maximum number of people needing medical attention at any particular time, how much good would quarantine of patients do in reducing severity of the epidemic. These are some of the questions. There are many more questions. And in this context, as mathematicians, would like to ask questions ourselves. What good a mathematical modeling can do? Mathematical, so these are the general, and this is a very old subject. Mathematical modeling in epidemiology provides understanding of the underlying mechanisms that influence the spread of the disease. In the process, it suggests important control strategy. Even Professor Anuj was very much emphatic on this, what a mathematical model will do. So in fact, this long history, so this mathematical, so these are basically called compartment models. So the population is divided into several groups, and each group is called a compartment. And mathematical modeling using ODE, ordinate differential equation, consists of describing the dynamics of movement from one compartment to another one. And this model starts transmission of a disease by a series of 3 papers by Kermack and McKendrick in 1927, 1932, and 1933. These are all published in Proceedings of Royal Society of London. So I will give you references later. Okay, so this is the modeling also often identifies behaviors that are unclear in experimental data, often because data are non-reproducible, especially in a health situation like COVID-19. So whatever happens yesterday, we cannot reproduce today. Okay, so that's the data. Whatever data we get, and the number of data obviously are limited and subject to errors in measurement. So this was also emphasized by Anuj, because the health personnel are under severe pressure, and so there's the data they're collecting obviously subject to errors. So most mathematical models in epidemiology, like SIR model, Professor Anuj was telling, and some other models also, they exhibit a kind of threshold behavior. So you are talking of this number also. If the average number of secondary infections caused by an average infective is less than 1, then a disease will disappear. We'll see it in differential equations. Okay, so these are not just words, but the model suggests this. While if it exceeds 1, there will be an epidemic. This number is called the basic reproduction. So he was talking of so many reproductive numbers, but this is the basic reproductive number, which I have denoted here by RC. But so Professor Anuj, for example, was using R0. So in the literature, you also find that notation R. Okay, so how that comes, we will see that. Okay, so going to mathematical models. So I start with okay, this different groups of population are put into different compartments, and the dynamics of the movement of one population from one compartment to another is described. So that is the mathematical model, general. So there are important assumptions. I will come to that, and especially in the context of COVID-19, I would like to comment on those things. So this is what I was mentioning. The basic compartment models that describe the transmission of communicable diseases are contained in a series of 3 papers by Kermack and McKendrick in 1927, 32, 30. So a special case of these works, okay, is the simplest model. This SIR model is the very basic and very simple and very crude also. You can see that. Okay. So we should also remember that the modeling process also depends very much on the strategies adopted by the health personnel and local government in containing the spread of the disease. For example, in COVID-19, we see this quarantining and isolating the population. So ideally, so there are other kinds of modeling, not just on ordinary differential equations. Okay, so there ideally, when a person is infected, that person may potentially spread the disease to others only after a few days. So this is called that incubation period. Okay, but here, using ordinary differential equations, we assume that he is potentially able to spread the disease instantly. Okay, then that may not be, and that leads to instead of ordinary differential equations, we end up in delay differential equations, and these are more technical and more difficult. And with ODE, we are able to draw some conclusions, but with delay differential equations, that may be very difficult, and we may end up only doing numerical computations. Okay, and there are several statistical models which I am not aware of, but there are many books and lots of literature using statistical models. So now I come to the basic, excuse me. So let me explain this basic SIR model. So Professor Anuj also describing that. So the whole population is divided into 3 compartments. So this S is the population that is susceptible, and I is the infected, and R is the removed population. Okay, so so this is the basic. So susceptible person. What is that population? A person in this class gets infected once she or he comes in contact with an infected person. Okay, so that's the assumption. I is the infected population, of course. Then R is removed population. So those persons having full immunity to the disease are isolated with vaccine, or it also includes the people who have died of the infection. Okay, if need be, so this class can be divided into 3, and but that in this simplest model that doesn't affect the analysis, as we see. Okay, so these are the, so please keep these terminologies into. So here I would like to add, so even in the simplest model, so the one main assumption is that, okay, maybe I'll just go to next slide. Okay. This is the important assumption. The total population is S plus I plus R. So sum of the populations in different compartments, and so I have put it in a different color here. So N remains a constant. Okay, so there is no migration to or from other populations, including births and natural deaths. Okay, so this last assumption may be reasonable because we expect the duration of the disease or pandemic is very small compared to the usual period required for deaths and births in the population. Okay, so that, but of course, if the disease remains for a longer time, again one has to change this assumption. Okay, and another phenomena we are observing in this COVID-19 is that, so even if we restrict to a locality in a city or a part of a city, because of the current developments, we cannot assume population that local area is a constant, because there is constant movement of people coming from other regions into this. Okay, how to model that? Okay, so that really creates a complication, and that the modelers have to address. So we are no longer satisfied with this constancy of this population. Okay, population itself is changing, and so we have to introduce a parameter for the growth. Growth is okay, but this, how to add the population from other regions, that's creating a lot of problems. And how many are infected there, how many are healthy, we don't know. Okay, so this, so for the, as far as this SIR model is concerned, so you keep this in mind. And the system of differential equations is very simple. Okay, so as usual, we denote prime by the differentiation with respect to time variable, and the system of equations is given by S prime equal to minus beta Si, I prime equal to beta Si minus alpha I, and this is the, okay, so this so this, the first equation describes rate at which the susceptible will become infected. So that's why that minus sign. Okay, the susceptible class will move into the infected class. Okay, if you look at the susceptible class will move to infected class. Okay, so that's what that describes, and beta is the rate at which the susceptible class gets infected. So that's called transmission rate. So that essentially means an average infected person, how many people he will meet in a period of time. It could be a day, it could be a week. So depending on that. Okay. And this from infected class, so this is increased from the susceptible class, so that same rate. And the infected person will get some treatment, okay, if there is some medicine, okay, or isolation, whatever it may be, and he is removed from that class. So that is the rate. So this is removal rate. Alpha is the removal rate, and then you get this R prime equal to alpha. So if you see the system of equations, this R prime, R is essentially decoupled from. So this is from mathematical point of view. So I'm just explaining the mathematics. So there is no coupling of this R. So once we know I, you just integrate and you get I. So essentially, it's a two-dimensional model as far as mathematics is concerned. So it's SI plane. Okay. So there is a reason for this writing in this fashion. So initially, if S is S0 at time t equal to 0, so because S prime is always negative, so S will be always less than S naught. Okay. So if I S is always less than S naught, so this term in the bracket is always less than or equal to beta S naught minus alpha. Okay. So if that beta S naught minus alpha is less than 0, we immediately see that I exponentially decreases. Okay. And that beta S0 minus alpha essentially gives you the basic reproduction rate is less than 1. So that's what Professor Anuj was also telling. So if that basic reproduction number is less than 1, the infection will die very quickly. In fact, there is no pandemic. There is no pandemic. But whereas if that is positive, that is greater than 1, then we see that I initially increases exponentially and then it decreases. So beta is the infection rate and alpha is the removal rate. Yes. So as I was explaining, this system is essentially two-dimensional. Okay. So if S is less than alpha, so again, go back to the here, you see that, just see the second equation in the system. The infected population decays exponentially and there is no pandemic. If S is bigger than alpha beta, there is pandemic. I initially rises exponentially but decays to 0 because S is always decreasing. So eventually S will become less than alpha by beta, and I start decreasing. Okay. And we can also see that number RC coming into play. So if we linearize around the disease-free equilibrium, so this is the total population, no infectives, and hence no no question of that removal. And then the second equation is just I prime equal to minus 1 by RCI, and we see that this again that same phenomenon. We can, one important mathematical thing here. Okay, so the analysis can be done in the SI plane. Let me show you that. You just forget the R, and that can be integrated. Okay, so we get this. So these describe the orbits in the IS plane. So C comes from the initial data. So for different initial data, you start IS0, I0, and you get a C here, and then you draw these curves in the IS plane, and those describe the orbits. Okay. So one thing we can immediately get is, so what about those orbits? So if you I have not drawn that, sorry for that. So if you draw the positive quadrant in IS plane, okay. So this simple, so the I and S are always non-negative numbers. So this is a non-negative number, and that very crude estimate you get S is always bigger than exponential of minus C by beta alpha, and I plus S is also less than or equal to the total population, and that gives you an upper bound for S. The important thing here is this lower bound, and this is always positive. This is always positive. Okay. So this orbits in the IS plane, S will never become 0. That's very, very important. And that means the orbits stay away from the I axis. S will never become 0. So that's very important. Very important. And because of these bounds, and S is always a decreasing function because S prime is negative. Okay. So this limit exists. Okay. And this is the population that escape the disease. So these many number of people, they never get infected. Okay. And in all these epidemiological models, okay, obtaining this S infinity is an important point, and we would like to know how many people do not get infected. That's very important. Very important. And other thing we learn from this model, again this S. So if you add the first two equations in the system, you get S plus I prime is minus alpha I. So S plus I is a bounded non-negative decreasing function, and this implies its derivative goes to 0, and that implies this IT goes to 0. So the infected population eventually dies out. Okay, that's what is the model suggests. And now we'll try to get an information about S infinity. Okay, so that is the population that escaped the disease. Okay, so how to obtain that in terms of the data given. So you can also see the constancy of the total population from this model. Okay, let me just show you that. If you add all these three equations, S plus I plus R, and take the derivative, you will get 0. So if you add these two, this will be 0 and this will be 0, and that shows that the total population is constant. So in this model, that's part of that. Okay. And now if you just, so I have written here S plus I prime is minus alpha I. So you just integrate from 0 to infinity, and you get minus alpha I it dt equal to, so this is a derivative. So you evaluate at plus infinity and at 0, and at 0 it's the total population. To begin with, you give the data S0, how many are infected and how many, the rest is that in the class S, so that is total population. So you get that n minus S infinity is alpha it. So still we have to remove this integral because that will not give you S infinity. So you have to remove this it, and again that is very simple. So again you go to the first equation. So you divide by S, so you get S prime by S equal to minus beta I, and now you integrate this from 0 to infinity. Okay, and the left hand side is just log S. The integration is log S, and you evaluate at infinity and at t equal to 0, and you get log of S0 by S0 by S infinity is equal to beta this integral. But this integral is n minus S infinity by alpha. And again, you simplify so that RC already introduced. So you get this. And this equation, maybe I have a name here. Okay. So this relation. So now you see, this involves the population that escapes S infinity, S0, the initial population who are not infected. That is S0, that we know. And RC we know from the modeling that alpha beta come from the observations. So we know RC, we know the total population. So this is called final size relation. Okay, so this is called final size. So that is the final size. So if you want to call this final size of the population, so this gives you a relation for that. So of course this is a nonlinear equation with the logarithm, and but it's very easy to see that it has a unique positive solution. Okay, so I have just shown you graphically here. So you draw that straight line coming from the right hand side. So this is the right hand. So this is just a straight line. So you replace this S unit by S. Okay, so you draw that line, and this is the logarithm curve. So starting from S0 to come down. Okay, the other things are irrelevant. So there are negative zeros, but we are not interested in that. So there is a unique positive. Okay, as you see here, you see if RC is very, very small. Okay, this S0, this S infinity is very close to S0. So this RC is very, very small. Even this graphically we see that a large percentage of the population remains not infected. Okay, so that's, and if RC is large, obviously this infinity will move away from S0. Okay. This so this is solution of the final size. So as I said, this is a very, very simple model, and you can also say it's a very crude. So it is not giving any details of the population. For example, now we are also seeing in the case of COVID-19. Okay, so senior citizens, 65 and above, and children below the age of 10 are more susceptible. So is it advised even do this depending on the age? Okay, there are age related models, age related who consider the age of the population into account. Okay, of course that makes the system more complicated, and the analysis also more involved. Okay. But this is also very easy to numerically integrate these equations. Okay, if we use, for example, this Runge-Kotak kind of type of schemes, we can easily integrate and also get numerical numbers, not just. And one good thing about this S infinity, the final size population. So using, so at what time we can guess? We can guess that the infection has come under control. So when you solve by some numerical scheme, there are no closed form solution, and at a time when S of t is very close to S infinity, because this is the final size population. So if S t becomes very close to it, we can be sure the infection it's going to end soon. Okay, so that's another significance of this final size population. Okay, so the population we can get by getting the transmission rate and the removal rate. So it doesn't involve any other. So now we are going to describe another model. Okay? So this is similar to B.S.R.V model, but it also takes into account quarantine and isolation process in the B.S.R.V model. And this quarantining and isolation, they can be thought as control measures, control measures to contain the disease. Okay, and presently in the COVID-19 situation also, we are seeing these measures being taken by the local governments. Okay, so this, so in SIR model, so you add 3 more compartments. Okay, so this is exposed. I will say what that exposed population is, and then Q is for quarantine, and J is for isolation. So since we are using for infected, so I'm using this J for the isolation. Model is developed in 2004 by researchers Bimal and his co-authors. So again, I mentioned that. So that model was developed after the outbreak of SARS in 2002 and 2003, and this paper is published in 2004. Okay, and this is called ACQI-JR model. So when a new disease appears in a population where no vaccine is available, just like COVID-19 we are facing now, isolation of diagnosed infectives and quarantine of people who are suspected of having been infected, and that is done usually by tracing the contacts of diagnosed infectives, are the only control measures available. So if there was a medicine or there were medicines, perhaps this COVID-19 would have been contained long back. Okay, since it's a new disease and there are no medicines, so one has to develop new strategies to contain the disease. So let me just show you the flow chart, and then okay, so this is again susceptible population, and that goes into this exposed class, and then some are quarantined, some are infected, and among infected, some are isolated again from the quarantine class, some are put in isolation, and finally from these 2 classes you enter into removed class. Okay, again removed class means that people are cured and some people unfortunately dead. Okay, some unfortunate deaths also included here. Okay, so we have to describe the dynamics between various compartments. Okay, and that is done here. Okay, so there are some again terminologies. I'll just read out. So I'll just mention the. For more details, you can refer to the literature. Okay, so exposed population. Exposed members are maybe infective with infectivity reduced by a factor of F on E. See that beta is there. Now you multiply this by factor, so that will be reduced. So in many infectious diseases, so what is that exposed members, exposed class? In many infectious infectious diseases, there is an exposed period after the transmission of infection from susceptibles to potentially infective members. But before these potential infectives develop symptoms and can transmit infection. Okay, so that is the description of the exposed population. So to incorporate an exposed with mean exposed period 1 by KE, and an exposed class is introduced. Exposed members who are not isolated become infective at the rate KE. Okay, so these are the assumptions in which go into the and Q is the quarantine population and J is the isolated or hospitalized population. Yeah, so again, it's there is no detailed description, so just put in quarantine and put in isolation. So exposed members are quarantined at a proportional rate gamma Q. In practice, many susceptibles are also quarantined, but we ignore this in model. So that's why you don't see in that flow chart from S to Q, we are not doing that. If you do that thing, so you have to introduce one more equation. So again, quarantining is not perfect, but it reduced the contact rate by a factor of epsilon. So these are the assumptions. The effect of this assumption is that some susceptibles make fewer contacts than the model assumes. So infectives are diagnosed at a proportional rate gamma J per unit time and are isolated. Isolation is also imperfect, and there may be transmission of disease by isolated members with an infectivity rate epsilon J. So you put all this two more assumptions, and quarantine members are monitored, and when they develop symptoms at rate KQ, they are isolated immediately. Infectives leave the infective class at alpha I, and isolated members leave the isolated class at, they get cured. Okay, they get cured and they leave that class and they enter into removed class. So that's the one. So if some susceptibles are quarantined right away, so we'll also have an arrow here and one more equation. Okay, so that has to be incorporated. And the system of equations, see earlier we had SIR only 3, and now this system is 6 equations, and these constants beta is already there in the beginning itself, and now we have epsilon E, epsilon Q, epsilon J. Again, if you add all this S plus E plus Q plus I plus J plus R, and that is the total population, and you see that the total population is again a constant. Okay, so in this model, so this built-in model, so the total population is constant. Okay, so as I remarked earlier, so now in case of COVID-19, and that is not happening, and modelers have to think how to incorporate that into the model. So we are, for example, we can say this S1, E1, Q1, so with respect to one model, and another model is another population is coming into this area, how to incorporate those changes. And that's a difficult task as I see it. So the model before effecting the control measures are quarantining, isolation. The special case, so if you look at that, so if you take Q and J, no quarantining and no isolation, that is called the SCIR model. Okay, so that's also studied very much, and compared to the past pandemics. Okay, so they are very good recording of past pandemics. Okay, so that's called SCIR model, which is simpler to analyze. Okay, so this model which incorporates quarantining and isolation is an improvement over this SCIR. Okay. So the aim is again to show that S eventually goes to S infinity, the population that escapes the disease, and derive the final size relation, and you show that the exposed class and eventually the infected all die out. Okay. The final size relation is exactly same as 2.3. So as let me just recall that. Yeah, here it is. This is final size relation. But only thing is, what is RC now? Okay. Because now there are so many parameters. There are so many parameters around. So how RC will come out of this? It's not even clear, right? See, earlier it was very simple to analyze, but now we have six. Even if we leave this R equation, there are 5 equations. So it's difficult to guess. Okay. So if you do the phase space analysis, okay, so this complicated RC, again that basic reproduction number, to get an idea of this RC, what that RC is. Okay, so before we actually go and find out. So let's linearize. So this is again a common mathematical technique we use in analyzing differential equations. So we'll linearize the system of 6 equations around this disease-free equilibrium. So the total you take just the total population and all other things 0. Okay. So if you linearize, essentially we need variables E, Q, I, J. Okay, S is equal to N, so that's removed. So essentially we get a 4x4 linear system, and the linear system involving this E, Q, I, J has this coefficient matrix. So it's a 4x4 matrix. 4x4 matrix. And as soon as a linear system, we look for the eigenvalues of this matrix and their nature. Okay, that's the usual analysis we do in differential equations. The characteristic polynomial of this is very difficult. So it's 4x4 matrix. So it's cumbersome. Okay, I'm not written it here. So what I mean by characteristic polynomial, I'm taking determinant of lambda I, the identity matrix, minus this matrix. Okay, so it's a 4x4 matrix. So it's a 4-degree polynomial with leading coefficient 1 and the constant term equal to a positive multiple of 1 minus RC, and RC is given by this expression. Okay. So that RC also comes as a coefficient in the characteristic polynomial and therefore plays an important role in the eigenvalues of the matrix. And as we know, the eigenvalues determine whether the solutions grow exponentially or decay exponentially. And we'll see an integration of the differential equation also comes out with the same number. Okay, so this is also hidden here. So this constant term is a positive multiple of this 1 minus RC. So if RC is less than 1, this constant term in the characteristic polynomial is positive. If RC is bigger than 1, the constant term is negative. And that determines, thus the constant term of the characteristic polynomial is positive if RC is less than 1 and negative is bigger than. And there are Ruth criteria for determining the nature of the eigenvalues of a polynomial. So one can show it's some work, it's more technical. So if RC is bigger than 1, there is a positive eigenvalue, okay, indicating an initial exponential growth rate of solutions of 3, 1. So there is a pandemic, okay. And if RC is less than 1, then all the eigenvalues have negative real parts, okay. Thus the solutions greatly decay exponentially because they all have negative real parts. Okay, so this is the usual linear analysis. Okay, again indicating that basic reproduction number, if it's bigger than 1, there is a pandemic. So there is initial spread of the disease and then it dies out. But if this basic reproduction number is less than 1, then there is no pandemic. The disease immediately dies. Okay, now the full analysis. Okay, so just I'll quickly finish. I know how much time I have. Excuse me.
10 minutes.
Okay, I just now the full analysis. So we take the initial conditions. T equal to 0, S0 equal to S0, E0 equal to E0, and all these other things. No quarantining. Initially. Only when the disease starts spreading, then we become non-zero. Okay, and this is the total population. So okay, that's important. Okay, and again, if you add the first 2 equations in the system of 6 equations, and argue as in the SIR model, okay, you immediately see that this integral 0 to t et, you remember, E the exposed class, okay, the population of exposed class. Et dt equal to n minus s infinity and ke plus gamma. Okay, again you can show that s goes to some s infinity and all other things go to 0. Okay, that's okay. And again, same analysis. Use the first equation, divide by s and integrate over 0 infinity, and that gives you log of s0 by s infinity as sum of integrals over 0 infinity of all these functions e, q, I, j. But we have only here integral of et. Okay. So using the other equations, okay, that's what I written. Using other equations in that system of 6 equation, it can be shown that the integrals over 0 in infinity of q, I, j all are equal to constant multiples of the integral of e. So whatever this log of s0 by s infinity involves all these integrals, integrals are in terms of only this integral et dt, integral of e. And when you put together all these things, okay, so you get again the final size relation, and this RC is precisely given by 3.3. Okay, let me again say that. So this is a big expression, very big expression. Oh. If you want to compare it to SIR model, so you put lots of things 0 here, so you get back that. So this basic reproduction number, okay, what we have learned here is it depends on the model we use. It is not the same number for all models. Okay, so it changes from model to model. That we should keep in mind. Okay. So okay. So you get this final size relation, and as we showed in the previous model, that it has a unique positive solution S infinity. So there is only one solution of this equation. Okay, so this also can be used, this RC. Okay, so the idea is to make RC less than 1 so that there is no pandemic, and as and when a vaccine or some medicine is available, so the strategy is to immunize a factor of the population, percentage of the population, so that you make this RC less than 1 so that there is no pandemic. And this is related to so-called herd community, which Professor Anuj Mubayi also was talking. Okay, so I am not going into that. So there are lots of topics one can keep on discussing. So here I have just discussed 2 simple models and discussed the mathematics behind that. And nowadays even common people are hearing about this R factor. Okay, so there is lots of talk of this R factor, and just explaining what that R factor is and how that influences the spread of the disease. Okay. Thank you for your attention, and I have listed here some references, and these, especially the first book, contains reference to many original articles on case studies, mathematical modeling and case studies. So even this book, there is lots of information you can find. So thank you. So if there are some questions.
Yes, sir. Thank you for your wonderful presentation, sir. Yeah, there are few questions.
Sure.
The first question is, can we use SAR model with the prey-predator model?
SAR model with
prey-predator model.
In fact, I forgot to mention that if you look at the these 2 models, they have so much common in that Lotka-Volterra model, right? They're very much similar to the predator-prey models. Of course, there is difference. So here the human population has become a prey and virus has become a predator. So that there is no, you can make it, you can add, but then the constancy of that n is gone. If you put a growth model in n, then the analysis becomes more complicated.
Yes, sir. Yes, sir. Sir, one more question. What is the benefit of considering delay differential equation model?
You are actually taking into account what's happening in real life. See, even when you inject a medicine in your body, it does not start reacting immediately. So there is a time delay. So even if, for example, in coronavirus case, we hear that a person, even who is infected, will require some 14 days of time.
Yes. To
spread the disease. So you are taking into account what's happening in real life. But that makes the mathematics problem more difficult and more job for the mathematicians. Of course, we can go for the numerical solution. But there are models even doing that. Okay. We may not have a neat expression for the final size relation and things like that. That we have to look in. Okay.
Okay, sir. Sir, Ashish Kumar, sir.
One question is, sir, because we can see in our country, labors are moving from one part of the country to another part. So can we take diffusion into account so that we can just closely model our to COVID-19 in our country? Because in our country, there is a movement of labor.
Sure,
Okay.
sure. Then, in fact, that is my big question now. So, see here in both the models, we have kept the population constant.
Yes.
And in reality, that is not happening. That is not happening. Now, the other population is also moving into this. How do you account for that? And what information do we have? Now, it is not an isolated population. It's not an isolated. See, in these two models, we see it's an isolated population. We are assuming that disease is only in that locality. Okay, but now there is movement. So how to incorporate that? How to incorporate that? That's a big task for modelers. And so we can, to begin with, we can just start with the two populations, one moving into another.
Sir, any more questions?
No, sir.
Thank you, sir, for giving nice lecture on compartment model.
Much.
And we learned a lot from this and how rigorous mathematics can play a role to just model COVID-19.
Yes.
So,
thank you very much.
Sir. Thank you, sir.
Thank you, sir.
Yeah. So I will mute my.
Okay, sir.
Yes, definitely.
I will mute. Okay, thank you. Thank you very much.
Sir, Ashish, sir, I'm sir, Bimal Kumar, Mishra, sir. Can you unmute yourself? I'm going to send you live. Ashish, sir.
Yes, sir.
You can give his introduction.
So next, Professor Vimal Kumar Mishra sir is going to give his presentation on transmission behavior of COVID-19. So as a professional experience, he did his MSc in Operation Research from University of Delhi and MSc in Mathematics from B.S.R.V University. He did his PhD from B.S.R.V University itself and DSc from Berhampur University. He served as a faculty member in BITS Pilani from 2001 to 2007 and in BIT Mishra from 2007 to 2017, and currently he is principal of Markham College of Commerce, Hazaribag. He is fellow of Royal Astronomical Society UK and Institute of Mathematics and Application UK. He is founder-in-chief of International Journal of Mathematical Modeling, Simulation and Application, and he is associate editor of International Journal of Information Technology and Computer Science and International Journal of Smart Computing and Information Technology. So I request Professor B.K. Mishra sir to just give his presentation on COVID-19. Okay, sir.
Yes, very good afternoon, Dr. Ashish and Dr. Prasad. And also, I was hearing Professor Datti. It was nice to hear him after a long interval of a time. And really, I also learned a lot from his lecture and more to explore on the recent work that what he just told in the last, that when the population is migrating, then how we can consider this. That is a big challenge to the modelers. In fact, it is very true also. And I will be just sharing these ideas. First, I will start with COVID-19 some of the history of the pandemic diseases, and later on the characteristic behavior of corona. And then I will come to the 3 stages. I have 3 different models on corona. And these models, what I'm going to present in front of you, is recently published in Chaos, Solitons and Fractals. And it's so in fact, when we speak about corona, so right from Anthony Plague, that was in 165 AD, the present coronavirus 2019 always have created some disaster to the humankind. And what I have the data with me as per the present day data, it says that around 216 countries.
Project your screen, sir.
When necessary, I will come to the screen.
Okay, sir.
Okay, then I will share it. Okay. So, as far as 216 countries, as per the latest data of 23rd June 2020 is considered, 216 countries having an infected cases of 91,92,000 with 4,75,000 cases of death, and it shows the disaster. Hello, are you able to hear me?
Yes, sir. Yes, sir. Yes, sir. We are able to hear you.
Yes, sir. Yes. It is audible.
Yeah, and as far as India is considered, till date, till morning, around 4,40,000 cases are positive infected, and 14,015 deaths are there, with a recovered rate of, in fact, 2,48,000 people have recovered from this specific disease. So in India, the recovery rate is pretty good. And in fact, how dangerous is this virus that we have to come into? We have to think this into consideration, how in fact this dangerous is. So the transmission rate are not the number of infected people from a single case, the case fatality rate, and whether we can determine that asymptomatic transmission is possible or not. These in fact determine the virus dangerous. So WHO estimated that R0 is between 1.4 and 2.5. That was in the very initial phase. But as per the latest data of WHO and as per the spread of the disease in the different part of the globe, R0 is estimated between 1.5 and 3.5. And as all of us know, since we are discussing on epidemiology, if R0 is less than 1, gradually the disease is going to disappear. But as far as we just compare this disease with SARS, and here in SARS, the common flu, the reproduction number was around 2. So on an average reproduction number when it was 2, so what we observe in the initial phases of China and the European countries and latest in US, the R0 in fact number has gradually increased, and it has started from 2.7 and now it is 3.12. So as far as this pandemic is concerned, so we try to understand that what in fact COVID-19 spreads. It primarily spreads through the droplets of saliva or discharge from the nose when an infected person coughs or sneezes. So that is one of the basic symptom, or that is what when we model a particular physical situation. So as a modeler, and all of us in fact try to understand the transmitting behavior, the biology, the physics behind the spread of the disease. So this plays an important role. As far as the pandemic disease is concerned, what I told in the very beginning, that the first recorded evidence of the pandemic was Anthony Plague. And in fact, it is also said to be plague of Galen. And as in fact, it spread, infected the Asia Minor, some portions of Egypt, Italy, Greece. And later on, it was termed as smallpox or measles. And the worst part of this pandemic was that it in fact killed over more than 5 million people across the globe. So it was one of the most disastrous pandemic in the history, human history. And then around in between many of the countries, small epidemic occurred. But the second evidence which occurred was in the 6th century, around 540 and so. So in fact, around 541, 542, there was plague of Justinian. And in fact, it was also said to be a bubonic plague. And in fact, it killed around 25 million people across the globe. So this, in fact, was one of the another disaster which happened to the humankind. And then we come to the middle part of the history. I don't want to go the entire history of the pandemic plague, but one of the latest of the plagues which occurred was scientifically said to be the flu pandemic, which had in fact also said to be the Asiatic flu or the Russian flu. And it was in the late of the 19th century. And it was thought to be an outbreak of influenza A virus subtype and H2N2, which is referred as. And this, in fact, was one of the latest of the epidemic disease, rather pandemic, which had occurred in recent time of the modern world. And since then, many of other cholera and plagues happened. But when we come across 100 years back, then between 1918 and 20, there was again a flu pandemic, and 25 million people across the globe died due to this, and 500 million people was infected by this disease. So it was one of in the 20th century, the early 20th century had this type of pandemic. And then HIV/AIDS came, that I don't want to discuss. But as far as the coronavirus history is concerned, so in fact, it has a history of around 50 years back. And in 1965, Tyrrell and Benoit, they found a virus passage named as B814. So this B814, it was in fact found in human embryonic tracheal organ cultures, and it was obtained from the respiratory tract of adult with a common cold. And then after that, 5 human coronaviruses appeared since 2003. And then, as per the latest 2002 to 2003 SARS emerged, that was a severe acute respiratory syndrome. And this also originated from the southern part of China and spread throughout the world. SARS epidemic, in fact, put the animal coronavirus in spotlight. That is very important, remarkable. So this SARS epidemic, when it was animal coronavirus, and in the last 20 years, there were several viral epidemics such as severe acute respiratory syndrome coronavirus, that is said to be the SARS, and H1N1 influenza in 2009. And latest in 2012, the other part was said to be the MERS coronavirus, that is the Middle East respiratory syndrome coronavirus, and it was first identified in Saudi Arabia in 2012. So we have to understand the history of coronavirus, where it originated and how it came into picture. And this SARS coronavirus, in fact, the death was not too much, and 800 death was recorded, which began in Saudi Arabia. And WHO, in fact, they very late, as far as this coronavirus is concerned, COVID-19, they very, I think that in the middle of February 2020, the Director General, in fact, he pronounced that this disease, in fact, is pandemic in nature. It was very late decision by WHO. So where this corona name appeared? So corona is a Latin word which means crown, and this crown is refers to its characteristic appearance, where its surface is covered in the form of club-shaped protein spikes, and therefore this name is, if you see the figure of coronavirus, so that will be very clear that it is appearing like a club-shaped. So everybody is just observing the figure of coronavirus and all. So this is a Latin word, what it means to say. So basically, when we speak about the transmission to humans, so therefore the bats to animals and animals to humans. But the surprising factor is that history, we don't have any scientific evidence till date that the coronavirus, the latest COVID-19, the transmission is in between the animals. There's a transmission in between the human beings. So back to humans, back to animals, animals to human was the transmission of this specific disease. So the basic reason why these animal coronavirus do not cause the symptom in the host reservoir, what I feel that they continuously co-evolve. These animals continuously co-evolve and adapt over a long period of time with the reservoir host. And therefore the symptoms are mild. And even if they show that it's not too much acute in nature, they're mild. And as far as you have to understand the symptoms of humans, birds, and animals, they have different symptoms. So if we just consider birds, the symptoms of this coronavirus in birds, it causes the upper respiratory tract infection. But while as the animals, it is considered, for example, in cows and pigs, it results in diarrhea. So, and in humans, it causes respiratory tract infection, and it would the common cold comes into picture. So, the high infection rate, the recombination rate, and the mutation rate of these animal coronavirus increases the probability of mutants' ability to transmit into another host. So, Dr. Prasad, while you are just putting a query to Professor Datti, was that whether prey-predator model can be taken into consideration or not. So, rightly pointed out by Professor was that it's a very good example by what we can use the prey-predator model here, because the symptoms we have to understand, because the infection rate, the recombination rate, the mutation rate of these animal coronavirus, they increases the probability of mutants' ability to transmit into another host. So prey-predator model, in fact, is going to play a very critical role for developing, for in fact understanding the transmitting behavior. But as far as we are very much concerned these days on the human population, because it's widely affected, and in the animals it is not so much. It's mild. Even we cannot understand that because the symptoms are for a long period of time, and with the reservoir host and they're mild, so we are not able to understand that in animals what type of this disease can happen. So prey predator, in fact, is going to play a very critical role when we develop a model and all. So the earlier form of the coronavirus was, I told, SARS and MERS were lethal, but they were not able to transmit in exponential manner, just what COVID-19 did. So that was a big question for all the scientific people. And what in fact are the symptoms? Because as far as the transmitting behavior is concerned, because the modelers always try to see that what is the transmitting behavior of these viruses in the human population. So we first of all try to understand the symptoms what we are having. So it is a respiratory disease, and most infected people develop mild to moderate symptoms, and they recover without having any specific treatment, because we don't have such a medical facility these days. We don't have vaccines, are still under research is going on to develop. So recovery without special, I'm speaking about the medicines which are going to be provided to the person who are infected by the disease. So that is not happening, but only the prevention, what Professor also told in the beginning, that they need the home isolation or the hospital quarantine, they have to take into consideration. So these special treatment are required for the recovery of this disease. But those having an age of 60 years and above and have a higher risk of developing severe diseases and death also. And the common symptoms which include is fever, tiredness, dry cough, maybe shortness of breath, aches and pains, maybe having throat infection, and very, very rare people, in fact, in country like Italy and some European part of the country have reported diarrhea, nausea, or runny nose. So in different geographical domain, we do the overall, if you see the population, the symptoms, then in fact it is just the breathing problem and the upper respiratory tract is being infected by this. So, but very few cases in Europe have reported for diarrhea for this specific disease. So, when we speak about the very first case of COVID-19, so all of us know that Yunnan seafood wholesale market of Wuhan, Republic of China, it was an animal to human transmission was in fact presumed as the main mechanism. Nevertheless, subsequent cases were not associated with this exposure mechanism. So we concluded the virus could be transmitted from human to human, and symptomatic people are the most frequent sources of COVID-19 spread. So why I told this because the Yunnan seafood wholesale market was in fact the primary place where the disease is spread. So animal to human transmission possibility was taken into consideration, and later on human to human and symptomatic peoples were considered for this. So when we in fact had the analysis for all those, so we do consider that what we had taken that the median time for exposure to the symptoms suggest that 14-day quarantine period. In fact, it was being used by the USCDS, that is the US Center for Disease Control. And they say that the 14-day quarantine period will be clear. And that also was in our paper. We have analyzed this with 97.5% of confidence that with 95% of confidence interval we have taken. So our analysis suggests that around 97.5% of the people who developed the symptoms of this coronavirus to SARS-CoV-2 infection will do so within 11.5 days of exposure. That was our percent of the people who have developed the symptoms. And for every 10,000 individuals quarantined for 14 days, only about 101 would develop symptoms after being released from quarantine. So that was a very good analysis. So it was stressed. It was stressed across the globe that more we quarantine, the more is the recovery, because that was the analysis from WHO also. So if we are going to isolate ourselves from the normal population from 14 days, then out of 10,000 individuals, only about 101 would develop symptoms after being released from quarantine. So it is a good recovery here. So we try to, I just want to share the slides right now. We have three different stages of COVID-19. So that's what I want to show. So the three stages of this COVID-19 that has the first stage, we have developed three different models based on these three different stages. So they are the first stage is the imported cases. I hope that we are able to see the slide. So the first stage is that imported cases where those who travel to other countries have contracted the infection. So three different stages, and we have developed models on these three different stages. I'm not going to discuss on the local and the global stability because the professor have already given about this analysis part. I will try to develop a model on these three different stages. The first stage is that imported cases where those who travel to other countries have contracted the infection. That means people where this disease in fact spread from China to Europe, China to Iran, China to US. So these were the foreign cases where the people in fact they started traveling from one source to the other destination, and the disease had spread after contacting those people. The second stage is the local transmission, in which people coming in immediate contact of an infected person report the infection. And third is the community transmission, when a person who has no travel history has contracted through domestic sources. And the fourth stage, that is an explosive stage. So there are several type of comments coming on from government also. And the latest report from ICMR director that India has not yet entered into community transmission, that is stage 3. But countries in US, many states united, and many of the European countries, in fact, have already entered the community transmission. But in India, as far as the official comment was there, there's no, India has not yet developed this community transmission. So, but when we speak about the imported cases, therefore first I come to the imported case. So what in fact is the case here? I here stress on the immigrant population.
Sir,
so immigrant population.
Your slides, we are not seeing the full screen, sir.
Okay, they are not in the full screen. Okay.
Not in a presentation mode.
So it's not in the full screen. Okay. So so are you able to see the full? Are you able to see the full screen?
No, sir. It's not. It's normal PowerPoint window only we are seeing, not the presentation mode.
Okay. So, however, I can make it into the full screen, but we are not able to see the full screen. Okay. So. So
check at the bottom.
Sir, you can. Sir, you can first go to. You can disable your sharing. First, go to full screen, then try to share the full screen window.
Sharing. I have made it into the full screen.
Yeah.
I have made it into the full screen.
Yeah. Now you'll see the two windows. You see the open the full screen window, sir. You can share the full screen window now. Presentation window.
Yeah, that is what I am making it. Huh? That is why I am making it like this only. So
yeah, now it came, sir. Yes.
Now you are able to see it?
Yeah, now we are able to see
yes,
it.
Sir.
Okay. Okay. So what in fact when I speak about immigrant population, I'm discussing on the first part on the second stage, the second and the first stage. We have taken into consideration a population said to be immigrant population. That it is the international movement of people to a destination country of which they are not natives or they do not possess citizenship in order to settle or reside this. Special permanent residents or naturalized citizens or to take up employment as a migrant worker or temporarily as a foreign worker. So here we have to understand we are not considering the case just which has happened during lockdown 4. In lockdown 4, the labels were immigrant there. So they were in fact traveling from one part of the country to the another part of the country, and the disease and some of them had positively infected laborers were there, and they in fact used to come to the other part of the country, and from there the disease is going to spread to the local residents. Here, in the very first stage, if you came from which were the person who were positively infected, so they were in touch with those cases. So here we are considering immigrant population for those population which are coming from the other country to our country, in which most of them or many of them were positively infected. So this is the case of immigrant population. This must not be considered for those population who are traversing the same country from Maharashtra, Gujarat, and from other people who are coming to the eastern part of the country. We are not taking into consideration those population. So in stage 1, we are considering this immigrant population like this. The second part is when we hear as the home isolation, that is the model in which we consider is the home isolation, that is Q1T. So as already discussed in the previous lecture, that isolation in fact plays a very important role. And in the 18th century, it was one of the first, when we had the plague in human population, the first medical treatment was of the home isolation. They were not home isolation, they were separated from the population. The population which are suffering from cholera, plague, they were in fact isolated from the population, and specific treatment was given to them. But here home isolation was that the restriction of the movement or the separation of well persons who are susceptible or possibly exposed to the contagious disease before it is known whether they will become ill. So isolation usually takes place in the home and may be applied at the individual level or to group of community susceptible or exposed population. So home isolation population we had considered in our model, in the very first model. So that is what, and the second is your, when we speak about that is the hospital quarantine. So hospital quarantine was also taken. So this hospital quarantine is the QT2 model, and this in fact is the restricted movement of the ill persons who have COVID-19, and in order to prevent its transmission to others. So it typically occurs in a hospital setting, but under very special cases may be done at home under a very special facility. Usually individuals are quarantined, but the practice may be applied in larger groups. So these two things we are well conversant with, that is the home isolation and hospital quarantine. So in lockdown 1, 2, 3, and 4, when we are speaking about these all 4 lockdowns. So till then we had hospital home quarantine, that is we were isolated from the population, the group of the population, restricted movement was there. So we did take this home isolation into picture, and those who were suffering, they were positively detected, they were subjected to hospital quarantine. And as already told that the recovered population is there, the population who recovered from COVID in fact 19. So when we are speaking about this, so what happens that these important part of the first stage, we did take into consideration all the 6 populations, that is the susceptible population, the immigrant population, the home isolation population, the hospital quarantine, the recovered population. So all these 6 classes were taken into consideration. So when we speak about these things, then what in fact this in the what happens that, as already discussed, that in the absence of vaccination and proper treatment, we had 2 control policies, the forced control policy named as home isolation and hospital quarantine, and these in fact across the globe were properly being followed. So this is what the diagrammatic representation of the first stage is considered. That is the imported cases where those who travel to other countries have contracted the infection. So if you do see, we have the susceptible population. this immigrant population E is playing a critical role. So these immigrant people, that is the population who has come from the foreign countries and in which a group of the population has positively infected. So the group of the people who were positively infected or those who were having the symptoms of COVID-19, they were subjected to the isolation, that is the forced isolation, home quarantine. And they are subjected to 14-day home isolation. And this immigrant population who, in fact, the symptom was not there too much, they mixed with this susceptible population. So, and also the susceptible, that when they came to their home or they were in fact subjected to having a contact between the other group of the population, that is the susceptible population. So this immigrant population, in fact, they were in touch with the normal population also, those who didn't show a clear-cut symptom. So in the very first stage, it was not so rigid. In fact, the rigidity was not there. And therefore, what we observed that the disease transmitted in the population, but it was not too much as what we observe these days. So the very first initial stage, they were in touch with the susceptible population. Those who were having some symptom, they were subjected to forced isolation, that is the home isolation. These home isolation people were also in touch with the susceptible population. So the susceptible population, in fact, due to this immigrant population, they were in in close touch with the immigrant population, and at a rate alpha 1, this immigrant population, a proportion of the immigrant population was in contact with the susceptible population. And this immigrant population were home isolated with a rate alpha 2. So a proportion of the population of the immigrant was in fact home isolated. And the home isolation population, that particular people or the group of the person who are home isolated with a rate epsilon, they in fact were in touch with the susceptible population. So what happened? This susceptible population, a portion of those immigrant and the home isolated people were in touch, and they in fact with a rate beta, they became infected. A group of them became infected. A proportion of the susceptible population became infectious due to these contacts. And the home isolation Q1 population at a rate delta, when they were having a positive symptom, or those who were declared that no, they require hospital treatment, having some breathing problems, having some other serious acute problems, they were diagnosed and they were subjected to, they were in fact infectious, highly infectious, and they were subjected to hospital quarantine Q2 at a rate eta. So this was in fact the Q2 population. And the immigrant population, people who were traveling from the other part of the country, they were in fact showing the positive nature of the symptom, who were positively infected, and though were showing acute problem, they were directly subjected to hospital quarantine at a rate 1 minus alpha 1 minus alpha 2. So this in fact was the hospital quarantine. So the immigrant population E, those who are having the acute problem, showing positive symptoms, were subjected to hospital quarantine directly Q2. But those who were having a mild type of seriousness, they were subjected to home isolation for 14 days. But the immigrant people who in fact was not showing any symptom, though it was a hidden, means it was not totally reflected, so they in fact mixed with the susceptible population. These were the initial cases just during lockdown 1 or just at the very beginning before lockdown 1 also. So And these home quarantine people also were in close touch with the susceptible group of people. So these susceptible group of people, due to the contact with these home isolated people, they were having if they had shown some symptom, they were infectious, and then they finally were the group of those people at a rate eta, they were subjected to hospital quarantine. And with the proper medication, I can say I cannot use the word medication right now because no vaccine is developed yet. But due to the ventilation or due to their total isolation, total preventive measures, what they have taken into consideration, these proportion of the population were recovered. So here we do consider two types of deaths. One is the natural death rate, that is mu1, that is being taken into consideration both in all the three cases, then the three compartments, that is the S compartment, E compartment, and Q1 compartment. They were all natural death. Somebody dies in that specific compartment, that is in the susceptible population, immigrant population, or the home isolation population, the death were natural in nature. But once they get infected or they were subjected to hospital quarantine, then there may be a natural death or there may be death due to COVID-19. That death rate was considered as mu2. And finally, when a proportion of the population got recovered, what we find in case in India, where 50% or 51% of the cases are recovered. So this particular recovery, the natural death rate is mu1. So this is in fact the whole model, what we have taken into stage 1. That is the imported cases. And then we have the differential equation for this. But before we have the differential equation, we do consider here two different types of birth rate. One is the natural birth death in the susceptible population, which we have added, that is B. And another what we have taken into consideration, that is A. A is again the birth of the immigrant population. Many people are arriving towards this particular population. The addition is there. So that positive case we have taken here, and that we have considered as A here. In the very initial phase of the COVID-19, there were reports that specifically in country in Italy and Spain, that a pregnant woman who was suffering from COVID-19 that had given birth to a child who in fact also suffered from COVID-19. But there was also a lot of press reporting and many other things, but there is no scientific evidence till date about the vertical transmission of the disease. That is, a pregnant woman suffering from the disease, the birth, the newborn baby, in fact, they are not in fact infected by the disease. There is no vertical transmission, which is scientifically established as per the latest report. But if the preventive measures are not taken for the newborn baby, then the disease may be contracted. But there is no genetic evidence right till date about the vertical transmission from one generation to the next generation. So as far as the newborn baby is considered. So we did consider here the birth in the susceptible population and the birth in the immigrant population. And thus we have the system of the equations for this. We have 6 classes. That is, we have the rate of susceptibility, rate of the immigrant population, rate of infectivity, rate of the home isolation population. We have dq2 by dt, that is the hospital quarantine, and rate of recovery. So when in fact we do consider this system of OD, what we have observed. So on the basis of all these, because equilibrium conditions and when we try to find the production number from the Jacobian matrix, so it is a 6x6 matrix, and based on all this calculation, we are able to find out the reproduction number to be like this. So I'm not going to go into the Jacobian matrix and all, because Professor already had explained in his beginning how to get the reproduction number for this case. So on the basis of this reproduction number, a very important parameter for epidemics, if it is less than 1, that disease dies out. and greater than 1, then in fact it is epidemic or the disease is there. So in country like India, in the present scenario, the reproduction number in fact has escalated to 2.85 as per the latest scientific evidence. So it may even cross 3 also, because the what we observe that the immigration of the population is there, and it is a big challenge that when the population is mixing from the one part of the country to the other part of the country, then what type of models or what mixing population we should take into consideration is a big question. However, we will try our level best to take this into consideration. We are developing paper on it, and that will be on the 3 part, that is, we'll be using the deep learning and the artificial intelligence technique and also the mathematical model, and try to get a very good example, a very good mixing for this. So these are not what we have observed. I'm not going into detail, neither the local or the global stability what we have proved. However, we have used the geometrical approach to show the global stability. So this was the very first model, and then we come to the second model. The second model, in fact, is about the second stage. That is the local transmission, in which people coming in immediate contact of an infected person, they reported infection. So this is the local transmission. We didn't hear the immigrant population has arrived, but they are not going to mix with the susceptible population. Once they have come, that is the lockdown phase 2 was there, 2 and 3, and lockdown 2, 3, and some part of lockdown 4. However, when we did consider the cases of Maharashtra, Gujarat, and also in Delhi, then there in the latest report, what we are observing these days, however, the government has ruled out about the community transmission, but in many of the we can say in the slums areas, the community transmission possibility cannot be ruled out. So stage 3 in many of the US and European countries, stage 3 was the case, and possibility of ruling out totally in country like India for many of the states has to be again seriously thought over it. Because it is the case which is being reported day-to-day analysis is being what it is being coming out, is throwing light or reflection on stage 3, that is community transmission. However, officially it has been reported that there is no community transmission in country like India presently. Therefore, we do accept this because ICMR director has already given this statement 4 or 5 days back. So this is the second stage what we are having in lockdown. This had in fact started in country in lockdown 2, 3, and in lockdown 4 in the middle till middle, this stage 2 was was up to a particular restricted zone, but after means just before lockdown 4 and unlockdown 1, this had exploded like anything. So this is the second stage where local transmission was there, and the people in fact they were in immediate contact. So we did consider all the 6 population here, but the immigrant people who had come here were being subjected to forcefully home isolated for 14 days, and the corona warriors, they have played a critical role for minimizing the spread in the local population, and the whole area was sealed out if any positive cases were found out. So the corona warriors who were in contact with those infected people, they in fact got infected, and also people who were in touch with those positively infected cases, they were also subjected to that specific disease. So we did consider here the second stage, and what you do observe here in the second case, we did have all the 6 compartments here, but the immigrant population were directly subjected to home isolation forcibly, and this was Q1. They were home isolated with a rate alpha 2, and they were not in contact with the normal population. Once they once they in fact had entered the country, they were subjected to home isolation. And then those who are showing acute case, those who are showing positive cases, they got infected. A proportion of the population of the home isolation at a rate delta, they were subjected to infectious cases, and they were subjected to hospital quarantine at a rate eta. So the exposed for the immigrant population were not directly subjected to, were not mixing with the normal population. They had, if you do remember, there were stamps put in their hand that they are home quarantine. That means they are not subjected to get themselves mixed with the susceptible population, which was in the first case. So this is the local transmission. And this, in fact, the corona warriors, that is the susceptible corona warriors, they were in fact in touch, the doctors, the nurses, or the hospital people, they were in touch with these home quarantine or the people who were forcibly isolated in the quarantine center. So in the quarantine center, many of them were those who were the people who were supposed to take care of these quarantine centers. So they were exposed towards that particular diseases and got infected with a rate beta. And then those who are having positive symptom and those who were acute, those who are having acute cases were subjected to hospital quarantine at a rate eta. But now the policies have changed. 14 days they were subjected to home quarantine, and but nowadays we just when we go through the reports, then they say that 5 days they are subjected to quarantine center, and if they show mild type of symptoms, then they can in fact directly go to home. So this explosion is spread, which I want to stress very much. So if you do not, because what is happening these days, when we hear or when we go through the evidence, scientific evidence, and also through the media reports which are coming out. So what we observe, 5 days only the quarantine is there. Somewhere they say, no, you go to home isolation, and they are subjected to, I'm speaking about unlockdown phase 1. So, and even in the lockdown phase 4 in the last particular phase. So hospital quarantine was not made these days when cities like Delhi, Mumbai, so these are having hospital crowding there. So they are only admitting those people who are severe in nature, and other people, they are going towards their home, and also in quarantine center only 5 days. So the exposure, the transmission of the disease, they are not making preventive measures, precautionary measures are not well taken into account, and the disease is spreading like anything in the population. So, and this is going to lead to community transmission number 3. So, stage number 3. So, this is a very acute phase what we are living these days. So, the prediction what we have forecasted using some test, some non-parametric hypothesis we have taken due to lack of some data in our country. We have used the Kruskal-Wallis test, and on the basis of this, we are able to say that we are able to predict that in country like India, where just after phase 4 and in the lockdown particular area, when this unlockdown has happened and the people are merging with each other in the susceptible population without having a home quarantine of 14 days, and they are subject, they're exposing the disease, and death in fact, and the positive cases may even lead to 4.85 lakhs. So that is a finding. However, we are not going to here expose this. So this is the second stage model where the immigrant population is not in direct touch with the susceptible population. They are going to home quarantine, and in this home quarantine, they are in touch with the corona warriors and many other hospital people, the police personnel which are taking care of the quarantine center, they get in touch with these home isolation people, and they are in the quarantine center. They are subjected to positive infection, and then with the rate eta, if the severe disease occur, if the acute symptoms occur, they are subjected to hospital quarantine Q2, and the rate of recovery here again is R, which is in India around 50.25%. So with the rate gamma, they are being recovered. So this is the second stage what we have modeled, and this again we have published this paper in Chaos, Solitons and Fractals. That is the 3 quarantine models. The paper name is 3 quarantine models for COVID-19. So based on this, we have the system of OD again, and this is the what we have found out. This on the basis of the 6x6 matrix, we are able to find out the reproduction number using a B.S.R.V method. And this reproduction number for this particular model is this. So now when we speak about the community transmission, that is the worst case in US and Italy, a very good example for the control of the transmission of COVID-19 across the globe was a country in South Korea and Japan, which had in fact taken a lot of preventive measures. Lockdown was in a very later stage in South Korea, but prevention was at a very highest level. So the transmission was not too much in that population. But in country like US and Europe, very specifically Italy, Spain, Germany, Britain, and specifically in country in Italy and Spain and in many of the states in USA, this community transmission was there. So community transmission rule out possibility cannot be done in Iran also, because that was also a disaster phase. Later on it was controlled. But this stage 3, we are at the verge of stage 3. Officially not, but the way in which the disease is transmitting in the population, and which the immigrant laborers are moving from one state to the other state, and preventive measures. And because if you see outside the world, if we just go to the market, we see that the prevention has not gone now. The fear has gone. People think that there is no COVID-19 now. So we are now at a community transmission these days. So community transmission, we are at the verge. And this is in fact a particular case what we want to prevent. But at the very same time, economics also of the country has to be taken into consideration. Therefore, this unlockdown one has happened, and maybe more relaxation is coming these days. But we cannot rule out the possibility of community transmission in a country like India. So it is the last stage where it can be controlled with great efforts. Otherwise, in the last stage, it is an uncontrolled epidemic disaster. That is what we have seen in US and many of the European countries. Russia is also in stage 2. Brazil has entered into stage number 3. India is at the verge of stage 3. So this, in fact, in stage 3, we are not considering any immigrant population. There is no international travel these days. However, only people who were outside, they were supposed to come in. So here we didn't consider immigrant population because here community transmission is there. And people do not have any travel history. Even if you are at your home, or if you have gone to market, you don't have any travel history. The disease may spread to you if proper prevention or precautionary measures are not taken. So immigrant population is no more present in this particular stage, and only community transmission is there, and people do not have any travel history, and they have contracted the disease through domestic sources. So many of the countries, they don't have any recent international travels. So when we come to this, the last stage, so disease is spreading from one person to another through direct contact, And in the absence of vaccination and proper treatment, we had already told that there are two force control policies, that is the home isolation and hospital quarantine. And in almost all parts of the country, to minimize the spread of the epidemics, these two force control are taken into account. So here we don't take into account the immigrant compartment. We leave it because this is the people who are suffering from COVID-19. They do not have travel history. They have contracted the disease through the infected people. And it's not necessary through whom I have got infection, that particular group or the individual person also do not have any travel history. So that is the part of stage 3 because it's already restricted. So this is what the model for stage 3 is. We have 5 compartments, that is susceptible, home isolation, the infectious class, the hospital quarantine, and the recovered cases. So proportion of the susceptible population goes to home isolation or the quarantine center, and they in fact they do not have any domestic sources, any positive symptom. If they are having, they are subjected to the infectious compartment. They are having a serious. We do consider the case in Delhi if we do take this particular scenario. So what is happening? People are subjected to home quarantine even if they are showing some symptom. They are positively having some breathing problem or having a fever. So they are subjected to they are subjected to infectious class. But those who are showing acute cases means high fever, high breathing problem, the lungs in fact are got choked. So they are subjected to hospital quarantine only. That is at a rate eta, they are going to hospital quarantine. And the rate of recovery is gamma here. And we do take here the two different type of the death rate, that is the natural death rate mu1 and the death due to disease, that is COVID-19 is mu2 also. So we see the death of rate, both the deaths natural and death due to disease in I and Q2 class. So we have this is stage 3. Good examples, what we can see in India is in Delhi, in Dharavi, in Mumbai, and some more places which is not yet reported. So we are at the verge. So, but it is but this case has occurred in Italy and in states. So that's what we have taken into consider. The data has been taken from that specific place, and accordingly it has been simulated. For this particular, we have the system of OD like this. And on the basis of this, we calculated the reproduction number. That was, and then we had used the geometrical approach, that is the Hub bifurcation, to establish the global stability for these models. And then we had simulated it. So we try to show you some of the simulated graphs for R0 greater than 1. And what we have observed, so we see the global stability for R0 greater than 1. That is in stage 1. It is well established here. The system is stable for different type of the trajectories. And what we come to stage number 2. So in stage number 2, if we see that this quarantine person population versus the recovered population. So if we see this particular graph, then in 11.85 days, this particular zone. So if we are quarantining it ourselves, then after 11.85 days, the recovery starts. And almost after 21 days, that is this particular case, after 21 days, what we observe, the recovery rate is very, very high. So there's a sharp increase in the recovery in stage number 2. So the 14-day quarantine, as per the data what we have observed from India, and we have made a comparative assessment with the other part of the most infected countries, we did observe that after 11.85 days, the recovery is starting, this particular phase. So that was for R naught greater than 1, and obviously we had about home isolation versus the recovered population. The more we are going to home isolate ourselves, the infection is very less, and the recovery what we see is very, very high. So home isolation, this is scientifically also established. The more we isolate, the more is the recovery. And also when we see about this particular graph, that is the susceptible versus the infection class in the SI model, this reproduction number greater than 1, we have taken here the reproduction number as 2.85. So our system is very much stable in nature. So that was all. We have these that what I was telling you about 1965 about Tyrrell and Noy. They were the people who, in fact, first of all, found the first generation of coronavirus, which was published in Lancet in 1966. So this is a very good paper in which we get the entire biological part and the transmitting behavior of the first COVID generation. So thank you so much for listening me patiently. Thank you so much.
Thank you, sir. Prasad, sir, do you have any question?
Hello. Hello.
Yes, sir, you are audible.
Huh.
So is there any question from DK Mishra, sir?
Yes, sir. How to calculate or not? Somebody asked how to calculate or not.
That is on Jacobian matrix we take into account, and already Professor Datti has explained, in detail what I found out in the last part of the lecture. That is Jacobian matrix. We have the negative eigenvalues, and as per that we find out this R naught. However, for 6x6 matrix, it is very difficult to find out analytically. So MATLABs, etc., can be used to find out R naught very simply for these all.
Okay. Yes, sir. That's all, sir. That is the only question from one question I received here. And if you have any questions, you can ask, sir. Ashish Kumar, sir.
I request Professor Datti to give us some advice on the future aspect of modeling this COVID-19, sir.
No, your talk was very good, and it explained lots of things, and you have done already a very good work. Very good work. My main question is how to incorporate different population into one locality. That you already done it. You have done it taking into account people came from abroad. But now how we track how many of those people in that population are infected or they are healthy. That kind of data. We can add more compartments. There is no problem with that. But how to get data? So that if we can manage, and maybe delay differential equations instead of ODE. We should look into delay differential equations.
Yes, sir.
Maybe they give a better picture. That's what I think.
What I feel that, sir, is that maybe these epidemic models, what we develop, already government has given a triple T scheme, but it is not effective. Triple T, that is testing, tracking, and tracing. We have taken this into consideration, but the data is not available due to these immigrant laborers which are coming.
Yeah,
so that is a big issue. So what we can do on that. Therefore, we have already submitted another paper to Chaos. We have used the deep learning.
As of now, treatment means quarantining or isolation, because there is no medicine or vaccine, right?
Right.
Obviously, the strategy will change once the vaccine comes into market. Then again, we have to incorporate in a different way.
Right, sir. Yeah. What I was telling that deep learning technique may be used. That is, artificial intelligence may be incorporated with these epidemic models, and the data, in fact, what we get from that will give us a good forecast. And also the different rates. So that is what we have done in our recent work.
As mathematicians, we will continue to do our job. Okay, I mean, if the parameters are like this, then that will happen. Certainly, we can forecast that. Numerical study is very difficult because we do not have a good, bounds for the what data you take, right? We should have a good. For that, maybe as you said, artificial intelligence can be used.
Yes, sir.
No, you want to run numerically, we need some numbers.
Sir.
We need some numbers, right?
So that is only possible if we are able to get the data from Arogya Setu app, as far as the country of India is concerned.
But
that may help us numerically.
Many doubts are raised about the data itself, right?
Right.
So we have to know, as Anuj was explaining, there is so much uncertainty.
Right, sir.
And demanding a good thing from mathematics is not fair when there is uncertainty in data, right? So, but we keep working, and hopefully you will have good data to finally validate. See, as Anuj was telling, we have to finally validate. That is the biggest challenge. Challenge. Right?
Yeah. Sir, one more question, sir. Can R not be calculated using Bayesian methods?
That if you go to statistical models. See, here this is a ODE model, right?
Yeah.
ODE model. So you just linearize around the disease-free equilibrium. And from the characteristic polynomial, you try to figure out the basic reproduction number. See, essentially, it's deciding the linearization. That matrix, when it has eigenvalues with negative real parts, and at least one positive eigenvalue. So that will decide the basic reproduction number.
Okay, sir. Thank you, sir.
So Kumar, sir. It was very, very good. Very nicely arranged. My first experience this online seminar. Now I'm very happy about this arrangement. It was really good.
Thank you, sir. Thank you, sir.
It was really good. So
thank you to Dr. Ashish and Dr. Prasad for
who managed it successfully.
Right. Very successful webinar. So many congratulations to them.
Because we are sitting at different parts of the country and even world, and yet managed to exchange our ideas, and very good discussion. And I hope the participants also would have gained some knowledge. Very nice, very nice.
Thank you, sir. Sir, Ashish sir, can you give just?
Yes, I request all participants to just to fill the Google form shared so that we can create certificates to them. So in question and answer section it is shared. So you just to fill up all the required names, emails, and their designation, everything, so that we can generate certificates. And thank you all, sir, for giving your valuable time to us. And it is nice to hear from all of you, and we gained so much of things, so that we can just in near future we can work in this epidemiology, because we have to just be ready for future challenges. And we have seen that how we can just go for mathematical model to understand the nature of epidemiology. Thank you, sir.
Thank you. Thank you.
Sir, thank you, Datti sir, and thank you, Bimal sir, for your wonderful lectures and enlightening us on this topic of modeling all these things.
A very good occasion. It is a very good occasion for us to show the power of differential equations. So ultimately, you know, that's power of differential equations that comes into play.
Correctly,
Okay,
sir. Thank you so much. Thank you so much.
Thank you all the participants for your participation. And now we are come to the end of this webinar. So have a nice time. Thank you. And we are ending the webinar. Thank you, one and all.
Thanks.
Thank you.